Okay, so it's 9:05 now. I understand that we're still expecting a lot more participants, but the workshop is recorded. So I think we'll move on now and
All
start
right.
with our presentation. And then if people join in later, they will always get the link to the recording afterwards so they can catch up in their own time.
All right. It's okay.
Okay, so my name is Alina Mushtaq, and I'm here with my colleague Louise, and together we represent the outreach team, and the Ensembl outreach team is responsible for giving and delivering workshops like these, and we are also responsible for managing the help desk. So if you have any specific questions related to your research or you have any feedback on Ensembl, and if you email help desk, we are the people behind it, and we also manage the social media pages for Ensembl. So to start with, I'm just going to talk about a little bit of the structure of this workshop. So what we're going to do is that we're going to have a presentation first, where I'm going to sort of give you background and talk to you about the data behind the tools that we have in Ensembl and how we produce them. And then we're going to move on to the Ensembl browser. So we're going to go to ensembl.org, and over there, I will show you how you can navigate the website or how you can use the tool. And after that, we're going to have some time for exercises, and I will show you where you can find these exercises as well in a minute. So these exercises are for you to sort of try and use the Ensembl browser by yourself and go beyond the demo a little bit. Please remember, it's not a test, so you don't have to send the answers back to us. It's just for you to try the exercises and sort of get familiar with the Ensembl data. So I'm going to show you where you can find the training page. So we can go to training.ensembl.org. So let me just go back actually. So this is training.ensembl.org. And once we go to this page, we can then on the bottom left-hand corner, we can find this tab that says Find Materials from Training. And this is what I'm going to click on. And from here, we will have our events, and then you can select the one specific to you. So this is Baze University. So I'm going to click on this. And on this page now, we have some course details. And then on the right-hand side, we have a PDF version of our presentation. So this will include my presentations and also what Louise is going to present. And then we also have a living document. So this is just a shared Google document. I'm going to show you what it looks like. So this document basically gives you a little introduction about the learning outcomes of this course. We then have a course overview, so everything that we're going to cover. And then we also have some space for questions. So I would encourage you, if you have any questions, to just type them out here. And then this document would just mean that you will sort of like have this database of questions and answers, and you can always save this document afterwards, so you can refer to it in the future. You can sort of refer to other people's questions and our answers to that. And it also means that it's quite nicely and tidily organized over here. Whereas in Zoom chat, if you ask questions and then somebody else has asked a question, it can get a little bit messy and we might miss a question. So what I'm going to do now is I'm going to share the link for this training page in the chat, so you can just navigate to it quickly. And then again, on there, you can find the PDF version of our presentation and also the link to the living document that you can use to ask questions. Then we have our, if I scroll down a bit, we have our section for demos and exercises. So I'm going to click on this first tab that says Species and Genome Assemblies. And over here, you can find our demo. So I will go through the demo on the screen, but if you want to follow the demo on your own, or if you want to do it in your own time, you also have that option, because over here we have nice screenshots that show you what everything is on the page. So basically it's everything that you will see us doing in the demo section as well. And then underneath it, we have this other tab that says Species and Genome Assemblies Exercises. And this is where you can find the exercises for different modules. So again, there are more exercises than we might have time for, so you can just pick and choose the ones that are most relevant to you. For every exercise, we also have a text answer. So if you're not sure about whether you've got the right answer, you can always refer to this. And then we also have a video answer where we just take you through the Ensembl browser and just show you on the screen how you can find the resolution to the question. Okay, so now I'm going to go back to the presentation, and we're going to talk a little bit about the introduction to Ensembl. So let me just go into presentation mode now. Okay, so we're going to talk a little bit about the history. So the first genome was first sequenced by Fred Sanger and his team in 1977. And it was a small bacteriophage genome of about 5,000 bases, which paved the way for sequencing of more complex organisms, including the draft for the of the human genome, which was published in early 2000s. It was a much larger genome of about 3 billion bases, and since then, many other organisms have been sequenced as well. The advances in the sequencing technology has now enabled the scientific community to sequence many different organisms, and now we have large consortia that are aiming to sequence all living things on Earth, such as Darwin Tree of Life, but it has also enabled sequencing of many different individuals of a given species. So, for example, different people of various ethnicities to assess the genetic diversity within the population and their allele frequencies. And Ensembl was started in 1999 when the human genome was in commission, as it was known that the automatic annotation would be essential. So Ensembl's goal was to automatically annotate the human genome and then integrate this annotation with other biological data and make it all publicly available. And since Ensembl's launch in 2000, many genomes have been added to Ensembl. So why do we need genome browsers like Ensembl? Genomes are big and they contain a lot of data, and we're producing them at a fairly massive rate. And if we look at the sequencing data, it can look like this, which is a string of letters of genetic code, and this data is completely meaningless without annotation. The only way this data will mean something is if we work with this data and we process it, and this is what genome browsers do. And Ensembl is a type of genome browser that takes publicly available sequencing data from genomes. It's important to remember that we don't do any sequencing ourselves. We take the publicly available sequencing data, and then we make sense of that data. So we annotate genomes with genomic features like genes, gene variants, enhancers, and we perform our own analysis on this data as well. So Ensembl is also called an added value resource. This just means that it brings together information from a wide range of other databases in a single site, and then we provide easy access to that data. We're not the only genome browser. We also have a sister page called Ensembl Genomes, which is dedicated to non-vertebrates. And then you might have also come across UCSC and also NCBI Genome Data Viewer. And we have a lot of data in Ensembl. So genome assemblies are the primary data type that we import from publicly available databases. And then we annotate these genomes with genes, with variation data. We have regulatory builds, and then we also perform our own comparative analysis on this data to have some data on alignments, we have gene trees, and also inferred homologs. We also have some tools to help you access the Ensembl data. We have BioMart, which is an online data export tool, and this allows you to export large amounts of Ensembl data without any programming knowledge. And Luis is going to talk to you about BioMart later on. We also have Variant Effect Predictor. So this is VEP, and this allows you to annotate your own variants. We also allow you to display your own data in the browser, and we provide programmatic access to our data via our APIs. And most importantly, Ensembl is completely open source, it's free to use, and we also provide access to our whole genome files stored in the FTP site. So I've already just hinted that there are many different access scales, so many different ways that you can access the Ensembl data. You can access the Ensembl data one by using the browser. And this is what we're going to focus on today. But you can also access our data in bulk using tools such as BioMart, Ensembl's VEP, also our APIs, or directly via MySQL. And as I mentioned, we also have our FTP site. So it's a public directory with whole genome flat files. And so these are basically large files if you wanted to download genome-wide data. Ensembl is dedicated to vertebrates, and there are about 300 vertebrate species, and the number is constantly growing. But you can also find non-vertebrate species, so you can find fly, so Drosophila, worm and yeast genomes, and these are there because these are very popular model organisms, and they're also there for some comparative purposes as well. We also have our sister site, so Ensembl Genomes, that is dedicated to non-vertebrates, and this is split by taxa. So you can find Ensembl bacteria, protists, fungi, metazoa, and plants on Ensembl Genomes. And they all follow the same logic and organization as the main Ensembl site. And here we have a comparison of Ensembl and Ensembl Genomes. So you can see that Ensembl is a lot older than Ensembl Genomes. And the biggest difference between the two is that we annotate most of the vertebrate genomes ourselves in Ensembl, whereas for the non-vertebrates, the annotation is done in collaboration with the scientific communities. So we would import the annotation from external sources, like for Drosophila, we would import it from Flybase. And Ensembl is updated every 3 months as genomic data constantly changes. So it's important to remember that our data is not static. Every 3 months, we release a new version, and behind the scenes, there is a lot of work as new genome assemblies are input, and we update our gene sets, we update our softwares. We also have new analysis. We run new analysis on the data, we update our variation and regulation data, and finally, we package it all up to then display it in the browser. So the current release is Ensembl 106, which was updated in April 2022. And then we're also working on Ensembl 107, which will be released sometime in early July. So this means that if you were working with Ensembl data and then you came back to it in 3 months' time or a few months' time, then the data there might not be the same. So it's important to remember the release number that you're working with. So if you wanted to keep your data consistent, you can always go back to that release number via our archives. And our archives are our older versions, which are kept frozen in time for up to 5 years. And I will show you where you can find the archives in the demonstration. And then we also have our rapid release site. So this is our site that has genomes that are released every 2 weeks. So this has genomes with gene annotations only. We also have BLAST supported here, and then we also have some homology predictions, but it doesn't have other tools like BioMart or web or any variation data. Okay, so let's talk about genome assemblies, which are the primary data type you can find in Ensembl, and how the genome assemblies are being generated. So again, I'm showing you a bit of a historical approach on how the first human genome was assembled. I need to start by stressing that currently there is no sequencing technique which is able to read the entire chromosome. Therefore, the DNA first has to be fragmented, and then the fragments are sequenced. So back in the day, step one was to cut the DNA into chunks, and then the human DNA was cloned into bags. So these are bacterial artificial chromosomes, which were then grown in bacterial cultures to amplify the number of DNA copies. And finally, the cloned DNA was sequenced from bags using Sanger sequencing. And later on, there was an era of PCR amplification. And nowadays, you don't really need any amplification at all. So sequencing creates these so-called sequencing reads of varying lengths, depending on the technology. But in all cases, there is a shortening of the genome, which is then matched up using overlaps to create a continuous sequence called contig. And here we have the terminology that we use associated with the genome assembly. So the set of sequenced bags cloned with overlaps plotted along the genome is called the tile path. So here we have the tile path. At each of these blue boxes is sequenced back clone, and you can see that there's some overlap between them and they are organized in order. So they're plotted along the genome, and then we have these back clones with trimmed overlaps. These are called contigs, and contigs are shown here as these blocks of different shades of blue, and then a set of contigs stuck together with no gaps is called a scaffold, and then we have one scaffold and then another joined together into a chromosome. I need to start by stressing also that human reference genome is not a genome of a single person, but at the same time, it's not a consensus genome either. So it's composed of chunks, and every chunk is real person sequence. So it's somehow a hybrid genome of different people's DNA. And people often assume that the genome assembly is a perfect representation of the genome, but I'm going to use this map analogy that you see on my slide to show you what it's really like. So if you imagine the genome, or all of the DNA within the cell corresponds to the Earth, the genome assembly would then be like a map with a coordinate system, with some annotations and some labels, and we would like the genome assembly to be as accurate a representation of the genome as possible. So something like we see now in Google Maps. However, in reality, it's something more like this. So it's not perfect. There are some gaps and errors, and there are some genomic regions that are difficult to sequence and reconstruct. There's also some repetitive stuff. So with time, the scientific community is filling in these gaps and correcting the errors, and the scientific community might issue some patches and some corrections. So the bottom line is that the genome assemblies do change and are being constantly improved upon. And then this brings me to the version of the human genome assembly in Ensembl. So the current human genome assembly that we use in Ensembl is GRCH38, which has fewer gaps in terms of base pair length, but also many rare and private alleles, which were previously found in the human reference, have been now replaced with more common alleles. Our GRCH38 data and software are regularly updated. We also have a dedicated site to the previous human genome assembly. So this is GRCH37. And this is still... Hi, is there a question? Okay. So we still also host the GRCH37. So this is the previous human genome assembly, which is still widely used in the clinical community, but please bear in mind that it has got some larger gaps. However, our GRCH 37 software is regularly updated, but the data there not so much. And then finally, we also host even the older human genome assembly, which is NCBI 36. However, it's quite imperfect and there are a lot of gaps and this is not updated. But you can find it frozen in time in our archives. Okay, so that was the introduction that I wanted to give you and a little bit of background that I wanted to talk about in Ensembl. And now we're going to move on to our demonstration part of the module. So I'm going to take you to the Ensembl browser homepage, and I'm going to show you how you can navigate the browser. We're also going to have a look at the species that we have in Ensembl and how you can find some more species-specific data. So let me jump out of my presentation. And I'm going to go to the Ensembl Genome browser. So this is ensembl.org. I'll share this on the chat as well. So I'm on the Ensembl homepage. Please do let me know if you would like me to zoom in or if you can see my screen properly. I think I'll just zoom in perhaps. Just one more. Okay. So we have our homepage here. To start with, we have this blue navigation bar at the top of the page, and this bar is going to be available on every Ensembl page. We have some of our most popular tools like BLAST, VEP, and BioMart. We also have this downloads text over here, which will take you to our FTP site where you can download whole genome files. We also have a link to our help and documentations. So you can find loads and loads of documentation pages on our tools and our data here, and also some video tutorials on how to use the tools. And then we also have a link to our blog. So our blog contains articles about any updates, so sort of our updates to the tools and the data with the different releases. We also post about the different jobs that are coming up in Ensembl. And also, if you want to get to know the Ensembl team a bit more, you can also find some articles on that. And then on the right-hand side, you'll notice that we have our, over here, some introduction about what Ensembl is. We have our release number. So as I mentioned, we are on release 106, which was released in April 2022. We have some bullet points on the data that was updated or included with this release. And then if I scroll further down, we have our link to the rapid release site. So again, this is a site that is dedicated to new assemblies with gene and protein annotation released every 2 weeks. And then we have some bullet points from our blog. So for example, you'll see we have a blog over here on the declarations. So what's coming in Ensembl release 107. So that's the future release for July. And then we also have some articles on the new data that we've introduced. And if I keep scrolling further down on the bottom of the page, you'll notice that I have this link over here that says View in Archive Site. So I'm going to click on this, and this is going to open a menu that is now going to show me all of the archives that are available. To start with, we have the Ensembl GRCH 37. So this is the previous human genome assembly. And then after that, we have our previous Ensembl releases starting from 105. And we have these until 2014. So Ensembl 75. And then over here, which is Ensembl 54, you can find our even previous human genome assembly, which is NCBI 36. So you can navigate to the different archives or the version that you were working with quite easily from here. So I'm going to jump out of this menu now and back to my home page. So I'm going to scroll back up. The next thing that I want to talk to you about are the search options. So you'll notice that we have 2 search options on this page. We have this search option on the right-hand side corner. And then we also have the search option in the center of the page, this blue section over here. So this center of the page search option allows you to search for a gene by its genomic coordinate, a variant, or even a phenotype, but this search is going to be species specific. So from this drop-down menu, you can select your species. So for example, we can select human, mouse, zebrafish, or any of the species that you are interested in that is available in Ensembl. And once we have selected our species from this drop-down menu, so for example, if I select human, I can then type in the human gene name that I'm interested in, coordinates, variant, or phenotype, and then it's going to give me results that are restricted to the human species. Whereas the corner search, so this right-hand corner search option is going to give you results across all species. So you can, again, type in here the gene name, the variant ID, the genomic coordinates that you're interested in, but it's going to give you search results across all species. And then if I scroll down now, we have this species section over here. And within this, we first have our sort of these favorite genomes. So by default, we have these as human, mouse, and zebrafish, but you can always change your favorite genomes to select the ones that you're interested in by clicking on this pencil. So this allows you to edit your favorites. But to do that, you will need to create an Ensembl account, which is completely free to create, and you don't get any correspondence from us. Then on the left-hand side, we also have a list, again, of all genomes that you can select from this drop-down menu. Or if you want to view the full list of all species, you can view them in a table format. So this is what I'm going to do now. So I'm going to click on View Full List of All Species. So this page lists all of the Ensembl species, and you can find the common name, but then you can also find the Latin, so the scientific name, and then you can find the taxon ID in the next column, the Ensembl assembly that we have, the accession number, the gene build method, and then we can also find some information on whether or not we have the variation and the regulation data available. So for example, if I scroll down for Algerian mouse, you'll notice that for regulation, we have a Y, which means yes. However, for variation, we have this hyphen. So this means that we don't have the variation data for this Algerian mouse species. You also have the option to download this table. So you can download the entire table, and you also have the option to filter this table. So for example, in this text box, I'm going to type in human, and this is then going to filter my table to show me human species. So which is over here. We have the common name, then we have the scientific name, Homo sapiens. We have the taxon ID, which is 9606. The current Ensembl assembly that we have, which is GRCH38, as I mentioned, the accession number for it, the gene build method, and then we also have indications on whether or not we have the variation data. Yes, we do. Or the regulation data. We also have the regulation data for human species. To find out more information about the human species, I'm now going to click on human, and this is going to then take me to the human species specific page. And over here, we first have the search bar. So you can search anything that you search for here is then going to give you results that are restricted to the human species. And then we have this section over here for genome assembly information. So we can find some more information and statistics. You can download the DNA sequence in the FASTA format. And then you also have some more example pages introducing different data types. But so we have comparative genomics, regulation, gene annotation, and variation. However, I'm going to focus on this section that is dedicated to genome assembly. And from here, I'm going to go to this more information and statistics hyperlink. So from this page, we can learn more about the genome assembly and gene annotation. You can find information about, so we have this summary statistics on the right-hand side, which includes the genome length. So we have the base pair length. We have this information on assembly provider. So it was provided by Genome Reference Consortium. The annotation providers was done by Ensembl. We have the information on the database version, the GenCode version, and then we also have some information on the gene counts in the primary assembly and also the alternative assembly. Okay, so this is everything that I wanted to show you on the main Ensembl site. I know that some of you are also working with non-vertebrates. So what I'm going to do now is that I'm going to open a new tab and let's just explore Ensembl genomes. So I'm going to type ensemblgenomes.org. So let me just share this page with you. So I'm going to share it on the chat. And as I mentioned, this is our sister site, which is dedicated to non-vertebrates. So on this site, you can also find our Ensembl COVID-19 browser. So this is a portal which hosts SARS-2 genome and annotation. And then you can also find a link to our rapid release site. And then you can find links to go to Ensembl sites that are dedicated to plants, metazoa, protists, fungi, and also bacteria. All right. So that was the introduction to the Ensembl browser pages that I wanted to give you. We now have some time for exercises. So we're going to go back to the training page. And we are going to focus on the exercises in this first tab. So this is species and genome assemblies. And if we just expand, so once you click on this species and genome assemblies, you'll see that we have these two tabs for demo. So this will show you everything that I've covered so far. And then we also have a tab for species and genome assemblies exercises. So we have some exercises on panda, zebrafish, mosquito, and also Ensembl bacteria. So just try and do the ones that are most relevant to you. And you can also find text answers and also video answers to the exercises. So I think we'll have about 10 minutes for the exercises and we will come back and then I'm going to show you the location tab. So how you can search for genomic coordinates and how you can view the different features that we have in the context of the genome. All right, so we'll have 10 minutes now for these exercises. And please remember, it's not a test, so you don't have to send the answers to us. It's just for you to try the exercises for yourself. Okay, so how are we getting on with the exercises? I hope that people were able to complete them. If there are any questions, please don't hesitate to type them into the living document, and then either me or Louise would be happy to get back to you. I'm now going to go on to the genomic regions demonstration. So we're going to go back to the Ensembl homepage. So this is on ensembl.org. And I'm now going to search for specific genomic coordinates. So these coordinates are mentioned in the on the training page in the demo section. So this is sort of just an example that I'm going to use to show you what the location of these genomic coordinates would look like and how you can view this location tab. So to start with, I'm going to focus on this center search bar, search option, because I want to search for specific genomic coordinates in the human species. So from here, I'm going to paste in my, oops, not this. not this. Okay, let's go back to the presentation, and from here, I have my genomic coordinates mentioned here. I'm going to copy these. And now I'm going to go back to the Ensembl page. And in the search section, I'm going to paste in my genomic coordinates. So I have my genomic coordinates here, but I have to define the species that I'm interested in because genomic coordinates are ambiguous. So we can find the same coordinates in mouse, for example, as well. So from this drop-down menu, let me choose human. Okay, so now we've chosen human, and the search results are going to show us the coordinate region, so the specific region for human only. The way that these coordinates are organized is that we first have the chromosome number, so it's 4 in this case. We then have a colon. After the colon, we have our start coordinate hyphen the end coordinate. So once you're happy with your species selection and your coordinates that you've typed in, we can just simply click on this Go button. And this takes us to the location tab. So this is the first tab that we're going to look at today. This is our region in detailed view. So Ensembl's data is organized in a tab system, and the location tab is the first tab that we're going to explore today. But then I'm also going to show you what the gene tab looks like, the transcript tab, and then Louise is going to later talk to you about the variation tab and the regulation tab. So basically we have tabs in Ensembl to show you the different Ensembl data. And then I also want to point you out to the page-specific help that we've got. So anywhere that you see this question mark on the page, so if you're in trouble, you get stuck, just always look out for this question mark symbol. This button is then going to open a page-specific help with a little bit of the explanation of everything that you see on the page. So we've got some screenshots with labels over here, and then in the left-hand section, we've also got some frequently asked questions, some video tutorials, glossary, and you also have a link to contact help desk. I'm going to close this for now. Then on the right-hand side, the left-hand side, we also have this menu. So this is always going to be page specific. So it lets you navigate through the page that you're looking at. And then after this section, we also have these buttons. So these are these blue buttons. So we first have configure this page, which allows you to modify and customize the view. We can add more data if you want as well, as by default. We don't show you all of the data that we have in Ensembl. We also have some options for custom tracks. You can also export the data. So I'm just going to show you what this looks like. So basically we have the location that we want to export over here. So this is our assembly, followed by our genomic coordinates that we provided. Then you can choose the output that you want. So you can have FASTA sequence, embed format, comma separated, tab separated, and other annotation file formats. Then you can also select the location. So you can change the chromosome number or the start and end coordinate. You can also change the strand orientation. So 1 would mean forward strand, -1 would mean reverse strand. Then we can also change the flanking sequence upstream and downstream. And then if you click on next, which is this blue button over here, you also get the option to choose your output format for the export. So you can export in HTML, text, or also compressed text. I'm not going to export anything for now. So I'm just going to close this menu either by clicking on this tick box or by just clicking on anywhere outside this box. All right, so on this page is composed of 3 views. We have our chromosome view, then we have our region in detail, and then we have our most zoomed in region in detail. So what you see on this page is that the 3 images are of different resolutions, and the next is more detailed than the last. So in our chromosome view, we have our specific region of interest shown here in this red box. And you'll also notice that we have this chromosome track over here. And then in the next view, you'll see that we have this chromosome band track, contigs track, genes track, and so on. So track is a genome browser specific term. It's basically a type of data plotted along the genome. So, for example, in this image, you'll see that we have this is our 1 megabyte view. So this image is centered around our region of interest. So our region of interest is still shown here in this red box. And this view allows you can also scroll along this, along the chromosome by clicking on these arrow buttons. You also have the option to sort of draw on this image. So for example, I can choose my mouse selection to this select a region option. And in this case, if I was going to click and drag my mouse across, I can then draw on this. And then I can either mark this region or I can jump to this region in more detail. But I'm not going to do that for now, but I just wanted to show you that you have the option to scroll across the chromosome or to select a specific region to zoom into it further. So the data here is organized as tracks. So we have one track for chromosome banding, then we have another track for the genome assembly. So this track is called contig, which is our double-stranded DNA, with contigs shown as these blue boxes of different shades. Then we have our genes track. So genes are shown as these boxes as well, and their names are shown below the gene, so slightly aligned to the left edge of this box. And then we have little arrows next to the gene names to basically indicate the gene orientation. And then we have our regulatory build, so our regulatory elements, and you can find some legend below that basically gives you information on what all of these colors mean on this image. And then if I scroll further down, we have our most detailed image on the bottom. So this is basically our entire red box. So it's only our region of interest, and you can see that there's quite a few different tracks over here. So there's many different tracks. We also have the genome assembly still. So we have our contig track over here. Genes on this image are shown as transcripts, and I'll talk about transcripts a bit more later on. So a gene in Ensembl is a collection of alternatively spliced transcripts, and you can see that gene is split into 2. So you'll see that we have the contig track over here, and then above the contig track, we have this gene track, and then we also have another gene track below the contig track. So all the transcripts, so these genes that you see above the contig track are the forward-stranded genes. Anything that you see below the contig track are the reverse-stranded genes. So another indication for this is that next to these transcript IDs, you'll see this arrow pointing forward. So this means that these transcripts have the forward orientation, whereas below you'll see that next to the transcript names or the transcript IDs, we have these arrows that are pointing in the reverse direction, indicating the transcript orientation. So as I mentioned before, that although there are a lot of tracks available on this image, we still have the option to add more tracks to this view. So let's manipulate and customize what we see here. So I'm going to scroll back up and I'm going to click on this blue button that says configure this page. And let's say that I want to add the UniProt track to my view. So the way that we can do this is we have the option to search for a track by entering our whatever we want in this text box. And then you'll notice we also have these tracks that are categorized by sequence and assembly, genes and transcripts. You can find tracks, so data tracks on mRNA and protein alignment, variation tracks, regulation tracks, comparative genomics related tracks, and so on. What the page shows me right now are all of the active tracks. So all of the tracks that are activated and are being shown on our page right now. I also wanted to mention that if you're not sure about what a certain track means, you can always click on this I button, which will give you more information about the track. So for example, for this Conti track, if I click on this I, it's then going to show you that this is a track that shows the underlying assembly contigs. All right. We want to add more tracks to our data. So I'm going to search for the UniProt track. And I have my search results here in this main display. And I have my UniProt track over here. So these are proteins from mammals from UniProt. So this is what I'm going to select. So to activate a track, you can just click on this box that's next to it. And I'm going to activate it as normal, but you do have the option to choose different track styles. So this is completely up to you how what you are more familiar with and what style you want. But I'm going to click, I'm going to turn it on as normal. Again, if you're not sure about what a track means, we always have this I button next to the track that is going to give you more information. So for example, this proteins from UniProt track is the mammalian proteins from the UniProt KB database that are positioned on the genome through BLAST-P of this GenScan predicted mammalian peptides to UniProt KB proteins. You also have the option to favorite this track by clicking on this star button. Okay, so this proteins from UniProt track is the only track that I'm going to activate for now. So once we're happy with our track selection, we can simply save and close this menu by clicking on this tick box. So now I'm going to wait for my track to load. It can take a little bit of time sometimes, depending on how busy the Ensembl sites are. Okay. So my image has now loaded, and let's now look for our UniProt track, which is over here. It's the third one in the line. So we've got our UniProt features over here. I also wanted to show you that you also have the option to sort of select this track and change its order. So if I just click on this track and then move it, I can change the order of this track. So I can bring it, for example, forward. I can bring it to the top. So for example, if you were interested in certain tracks and you wanted to have them at the top. All of these signs that you see on this image allow you, so this cogwheel allows you to configure this image. You have the option to add custom tracks. You can share this image with collaborators. You can resize the image. You can export this image. And then you also have the option to reset the configuration. So for example, if you've added loads of different data tracks, but you want to go to your original display, then you can just click on reset configuration, and it's going to take you to the default display. And then you can also reset track order. So again, if you've got loads of tracks that you've added to your image here, and then you've sort of repositioned them, but you want to go to the original track order, then you can just reset it from this button. Right. So this is everything that I wanted to show you on for this demonstration. And what we're going to do now is we're going to take a 5 minutes break. You can also sort of go to the training page, and the exercises for this module are going to be in this exploring genomic regions. And if you click on this exploring genomic regions, we have a few exercises mentioned over here. So I would encourage you to try them in your own time. So we're going to take a 5 minutes break for now, and then I'm going to come back and I'm going to talk to you about genes and transcripts. All right, so we're going to come back now, and I'm going to start with another demonstration. And with this demonstration, I'm going to focus on where you can find the gene tab and the transcript tab, and what sort of information you can get from these different tabs. So you can find some background information on genes and transcripts within the presentation. So if you go to the PDF presentation, you can find some information on there. But what I'm going to do is that I'm going to demo the genes and the transcripts data that we have. And then as we go through the data, I will give you a little bit of background about it as well. So I'm going to search for the human UQ-CERQ gene. So we'll type this in the chat as well, so you can look for it yourself as well. UQ-CERQ, human gene. So for this, we're going to focus on the center search section. From here, I'm going to select human. And now I'm going to type in my gene name, which is UQ-CERQ. And I'm now going to click on Go. So I've got my species selection, I've got my gene name here, and now I will click Go to search for this. And here I have my search results. So it's the first one that we're interested in. So this is UQ-CERQ human gene. And I'm going to click on this. And this takes me to the gene tab. So this tab collects information on the genes. And here we have the gene tab open, but you'll also notice that next to this gene tab, we also have the location tab, which opens automatically. And this shows you where the gene sits in the genome. So you can toggle between these two. And then we have this page-specific menu, which lists all of the available gene views. So we saw this before as well. And then on the bottom, we also have some tools to help you customize this page or add your own data or export this page. So I'm going to scroll back up and let's focus on this main view. So we have our main view that is composed of a little overview, which gives you the gene name, the Ensembl stable ID. Then we have the description of the gene, the synonyms, the gene location. So it's on chromosome 5. We have the coordinates and it's on the forward strand. We have some more information about this gene, that it has 6 transcripts, 207 orthologs, and it's associated with 3 phenotypes. And then we have this blue button that says Show Transcript Table, which you can choose to show or hide. And the transcript table summarizes all of the transcripts of this gene. Then we have in the first column the transcript ID. We have the transcript name, the base pair length of the transcript, the protein length, the biotype of the transcripts. They could be protein coding, they could be processed transcripts or retained introns as well. We have information on the CCDS and UniProt match, also the RefSeq match. And then if I scroll across more, we also have some flags. So these flags are basically an indication of the transcript quality. So people often ask us which transcript should they use to work with. So for example, if they want to design a CRISPR knockout, there is no straightforward answer to it. All of the transcripts are biologically valid, but we can provide some guidance on choosing the most representative transcript. So if I was to choose the most representative transcript, so the one to work with, I would first look for main select, which is available for humans only. And this is the most representative transcript. So it's most highly conserved, most well supported, highly expressed. And this transcript also matches the GRCH38, and it's 100% identical between RefSeq. So RefSeq is the NCBI transcript database. So it matches RefSeq, and it also matches Ensembl. So the first one would be that, and main select is only available for human. We also have Ensembl canonical. So Ensembl canonical flag is available for human, but also for all other species in Ensembl. So for humans, main select and Ensembl canonical are going to match each other because, again, Ensembl canonical is the flag. So this transcript is the most conserved, most highly expressed. It has the longest coding sequence, and it is also represented in other key sources such as NCBI and UniProt. So if for example, for your gene, you don't have the main select or Ensembl canonical available, then we would move on to APRI. So APRI is another transcript scoring algorithm, and this is developed by a group in Spain, and these guys use their own judgment to score transcripts, but by similar focus to us, but they also have some slightly different priorities. So they look at protein structure information, presence of active sites, cross-species alignments, and they score transcripts from 1 to 5, with 1 being the best. So P1 stands for principal isoform 1, and then we also have, for example, P2, but this is not shown here. But just remember that 1 would be the best and 5 would be not so good. If APRI is not available either, then we can move on to GenCode Basic. So GenCode Basic, we know that we make our transcript annotation based on real biological data, as I mentioned, and biological data might sometimes be incomplete. So if there are some fragmentary transcripts in our data, we would still make an annotation, but then we would flag these transcripts as incomplete or either missing at 5' or 3'. And GenCode Basic filters these incomplete transcripts out. If GenCode Basic was unavailable, then we would move on to transcript support level, which is this TSL, which is another scoring algorithm, and it also scores from 1 to 5, with 1 being best. Right. So if we look at, for example, this table, you'll notice that this first transcript that we have has the main select flag, it has the Ensembl canonical flag, it has the GenCode Basic flag, and also for APRIs and TSL, it has the 1 score, so it's the best. So this is the most biologically representative transcript for this gene. Okay, the next thing that I want to show you is the different gene-based displays that we have on the left-hand side. So if you want, you can view the sequence of this gene. So I'm going to click on sequence on the left-hand side. And I'm going to hide this transcript table for now. So we have the sequence of this gene here. So our we have our sequence of gene of interest, and then we also have its flanking region shown. So again, we also have our page-specific help. So basically, if you want to find out more information of everything that you see on this page, you can always click on this question mark symbol that will show you nicely labeled screenshots of everything that you see. Then we have the option to download this sequence, and you also have the option to blast this sequence. Underneath these buttons, we have this legend that tells you that the exons of our gene are shown in this brown lettering. And then all of the other exons in this region have this peach highlighting. So you'll notice that we have this peach highlighting with the black text, which basically means that these are exons of the neighboring gene. But then we have this bold brown highlighting, brown letter that's also highlighted in peach, but these are exons of our gene of interest. If I select a region, so if I click with my mouse and drag my mouse across and select a specific region, so a specific length, I then also get the option to blast the selected sequence. And I also wanted to show you the different ways that you could download this sequence. So if I click on Download Sequence button, you can then choose the file format. So you can choose from FASTA or rich text format. And then we also have the option for different settings. So the different sequence that you might want to export. So you can export cDNA sequence, coding sequence, amino acid sequence, 5' UTR, 3' UTR, exons only, or introns only, or genomic sequence. You can also select the 5' sequence. This is set to 600, but you can always shorten it, and also the 3' flanking sequence. And then this shows you what the file formats would look like. So we have the FASTA format over here, and then this is the rich text format over here, which basically it will show you the downloaded sequence with different it's a static image with different coloration and decorations, and this can be opened with a word processor. So I'm not going to download anything for now, so I'm just going to jump out of it. Okay. The next thing that I want to show you is we also have some information on gene ontologies. So these are the functions related to genes of our interest. So we have gene ontology for molecular function. This is grayed out here. So we might not have data on this, but for different genes, you can also find molecular function. We have gene ontology for biological process and also for cellular components. So let's click on biological process. So this is on the left-hand side in this ontology section. Hi, is there a question? Okay, they might have unmuted themselves accidentally. So this is what the gene ontology biological process table looks like. So we have the accession number, we have the gene ontology terms, the evidence for this gene ontology, the annotation source, and then we also have the transcript IDs that are associated with these gene ontologies. So please note that we have transcript IDs here, but not gene IDs. And this is because gene ontology terms are associated with transcripts or protein. And you'll notice that this specific table isn't very long, so it isn't very big, but these can get quite massive. So you can search for specific gene ontology terms or accession numbers by typing in this filter, and then you also have the option to download this table by clicking on this Excel button on the corner. Okay, so the next thing that I want to explore with you is external references. So also on the left-hand side, we're going to click on external references. So this shows us a table of what our gene corresponds, so the different databases that our gene corresponds to. So you can find information, so matching information from Expression Atlas, HGNC symbol, MIM, so this is from OMIM, NCBI, Reactome, and also Wikigene. And then we also have some more information on the transcripts of this gene and what databases they would correspond to. Right. So this is everything that you can find on the gene tab, and there is a lot more as well. So Louise is going to go through comparative genomics with you and also the variation data that we have. So you can always sort of play around with this gene-based displays on the left-hand side and find out specific gene-related information. The next thing that I want to show you is the transcript tab. So I'm going to click on this table that says Show Transcript Table. And from here, I'm going to click on this first transcript ID. So this is our most biologically representative transcript. And I know that because it has the main select flag, the Ensembl canonical flag, and then it also has the GenCode basic, APRI, and TSL scores of 1. So I'm going to click on this. And this is now going to take me to the transcript tab, which opens next to the gene tab. Again, we have a little overview. So this is the same overview that we've seen before for the gene tab as well. But then we have some additional information about this transcript. So this transcript has 3 exons, is annotated with 11 domains and features, and it is associated with 793 variant alleles. I'm going to hide this table to just make this page a bit tidier. We also have this nice graphical representation of this transcript. So you'll see that this transcript has these exons. So exons are shown as boxes. So we have 1, 2, and 3 exons. We also have some more information about the CCTS match, the UniProt match, the version of this transcript, the type of this transcript, so it's protein coding, the annotation method, and also the GenCode basic gene. So for the annotation method, the transcript, so the annotation method for this comes from the Havana manual annotation and also the automatic annotation. So in Ensembl, we have 2 annotation methods. We have the Havana manual annotation. So this is basically an annotator, so a person that looks for the evidence to make an annotation. And then we also have an automatic annotation, which is mostly computer-based. The Havana manual annotation is only done for human and mouse. And then for zebrafish and rat, it's done in most difficult regions. But please remember, Havana is mostly only done for human and mouse genes. All right. For our transcript-based displays, you can view different sequences of exons, cDNA, and you can also view the protein sequences. We also have some protein information in the form of protein summary, domains and features, and then we also have some information from PDB 3D protein models. So I'm just going to show you what this looks like. So I'm going to click on PDB 3D protein model. And this shows you a really nice 3D protein model from PDBE. So this is a nice interactive widget which shows you the 3D structure of the protein. And then you have the option, if I click on this, you can always rotate it. And then we also have the option to, okay, I'm going to zoom out a little bit to, here you go, to view the settings on the right-hand side because they were appearing at the bottom. Okay, so this menu on the right-hand side shows us that this is an Ensembl PDBE mapping. We have the protein ID for this over here. And then we also have the option to show exons. So we can click on this I button to show the exons. So we have, if I just expand this, we have exon 1 shown in purple. We have exon 2 shown in this green highlighting. And then we can also show some more protein information from Gene 3D, Panther, and PFAM. And then we also have the option to show SIFT variants. So you can see that I've just unhidden these by clicking on this eye, and we can see these variants on our protein model, and you can also show the polyphen models as well. Louise is going to talk more about SIFT and polyphen and variation data after this module. Okay. Another thing that I do quickly want to show you is the cDNA sequence. So as I mentioned, that within this transcript-based display, you can view the exon sequence, you can view the cDNA sequence, and then we also have the option to view the protein sequence. So I'm going to click on the cDNA sequence. So this is our sequence of our mature mRNA. And again, we have this question mark, which will show you, if you click on this, it's going to show, open a new page showing you everything that you see on this page. We also have the option to download this sequence or blast this sequence. So right now we have our sequence over here, and you'll see that it has these different highlightings on this. So these different highlightings are our variants, but I don't want to go into the variant information. So I'm going to remove this variant highlighting. And to do that, I'm going to go to configure this page on the left-hand side. And from here, you can choose the different display options. So you can choose to show exons only. You can choose to show codons, UTR, coding sequence, protein sequence, and you can also choose to show or unhide variants. Right now, this is activated, but I'm going to not show this by clicking on it. So I'm going to hide my variants. And now I'm going to save and close this menu by clicking on that tick box. And now my cDNA sequence is a lot neater. So you'll see that we have these 3 lines of sort of sequence shown here. And each of these lines means different things. So the first row is our actual cDNA. So this is the splice transcript with UTRs that are indicated by this yellow highlighting. Then our second row is our coding sequence. So it's CTS, and it starts with ATG. So we have A over here, and then T and G over here. And then the third row is actually our protein sequence. So again, it starts with a methionine, and then somewhere at the end, it's going to stop with this stop symbol. We have our codons mentioned over here in this alternative highlighting of white and light yellow. And then we have our exons mentioned in the black lettering, also as alternatives. So the first exon would be in this black lettering, and the start of the second exon would be in blue lettering, and then it will carry on like that. Again, if you're not sure, so if you're not, if my explanation isn't that good, we can always click on this question mark button over here, which will also show you. So it's basically going to show you what the different coloration on the page means, and then also what the different lines are. So it's telling you that the first line is the start of the cDNA, the second line is the start of the coding sequence, and the third line is the start of the protein sequence. So I'm going to close this for now. And this is everything that I wanted to show you on the transcript tab. But you can see that if you notice in these transcript-based displays, we have loads and loads of more information that you can play around with and sort of explore. So I hope that you've enjoyed looking at the location tab, the gene tab, and the transcript tab. And now I'm going to give you a 5 minutes break, and then we'll come back and we will talk about, Louise is going to talk to you about variation data. And please, if you have any questions, please do not hesitate to ask. Either if you have questions whilst I'm doing the demo and you don't understand something or you want me to go over something, please do unmute yourself and let us know. And similarly, if you have any questions specific to your research or you want some more explanation, please do send the questions in the living document. And I also wanted to mention that although I went through the different gene-based information and the transcript-based information throughout the demo, you can also find some more background information in the PDF presentation that you can find on the training page. So we have more information, for example, on canonical transcripts, the main select transcript flags, and gene ontology.
All right, hi everyone, welcome back. My name is Louise, for those who haven't seen me at the beginning. I will be covering the rest of the workshop. So yeah, let's start with the variation module now, and then I'll go through the variant effect predictor, which is a tool on Ensembl, and afterwards we'll take a lunch break. Okay, so let me know if you can't see my screen, or if you would like me to speak up, just put it down in the chat box, or just unmute yourself and let me know.
Screen is good. Thank you.
Okay, cool. Thank you, Alina. All right, so let's start with the variation module. In this module, I will talk about how variants are classified, the species for which variation data is available, and sources from which Ensembl retrieves data from. We will explore the variation data on Ensembl during the demonstration, and in the end, you will have some time for exercises. So let's talk about variation types. We can group variants according to their size or the number of nucleotides that are affected. Variations can come in a small scale where only one or a few nucleotides of a gene are affected. So here we have two sequences. The top sequence is the reference sequence, and the bottom sequence is your own data. So small scale variation types include small insertions and deletions, which are also called dips or indels, or these can be single nucleotide polymorphisms or SNPs. So these are changes in the nucleotide itself. In this example, the reference sequence has the A variant here, and our sequence down here has a T allele. Sorry, T allele or T variant. So variations can also come in a larger scale, such as in the chromosomal structure. These are also called structural variations, and the types of structural variation are copy number variants or CNVs. So for example, when you have a genomic sequence with a lot of repetitive regions, and there are also large scale deletions, duplications, insertions, and translocations. In Ensembl, there are 24 species that have variation data available. You can click on the link down here in the presentation to find out which species these are and the sources from which we take this data from. So where does the variation data come from in general? There are 4 steps in the Ensembl variation data process. So first, variation data is imported from publicly available data. During the import, the data is being checked for the quality, and then we compare the data that pass the quality control with other databases to link them with associated data or studies. And finally, we analyze the data and process them to make them available on Ensembl. So let's look at each step in more detail. In the first step, we import variation data from publicly available archives and sources. We search variation data from a number of different databases. So for human, for example, we retrieve data from dbSNP. For short genetic variations, we retrieve structural variation data from the database of Genomic Variants Archive, or DGV archive. For non-vertebrate species, we retrieve variation data from the European Variation Archive, or EVA, and we retrieve somatic variation data from COSMIC and variants implicated in cancer from the Cancer Gene Census. In the second step of the Ensembl variation process, we apply some quality control measures on the data that we've imported. So here we map the variants, sorry. So here we map the variants to a reference assembly. We compare the reported alleles with the reference genome and exclude any suspect variants. So for example, alleles that contain ambiguity codes or non-nucleotide characters. You can read more about the different quality measures that we apply in this link again on the presentation. And in the third step, we link the variation data that passed the quality control to other databases. So here we add value to the data, for example, by linking a variant to allele frequencies or phenotype or any publications. So allele frequencies are one of the main types of data that we link to our imported variation data. Allele frequencies describe how often an allele is present in a population. So for this, we link the imported data with databases such as the 1000 Genomes Project, NOMAD, UK10K, and TopMed. We also link the imported variation data with phenotype and disease information from a range of different sources depending on the species. So for example, for human, we link variation data to the human gene mutation database. Where is that? Oh, here it is. HGMD, and also ClinVar. And for mouse, we link variation data to the International Mouse Phenotyping Consortium, or IMPC, and we retrieve information such as the clinical significance of the variant, inheritance type, the reported gene, and risk allele. For genome-wide association studies, or GWAS, we also retrieve statistical information such as the p-value, odds ratio, and beta coefficient. In the final step of our Ensembl variation process, we apply our own analyses to the variation data that we imported and that have passed the quality control stage. So here we predict the variant consequence, protein function. We also calculate linkage disequilibrium and variant conservation across the different species. So how do we predict the variation consequences? Basically, we take each variant and we plot these along the genome. And we predict the functional consequence of the variant depending on where the variant falls in a gene. So where it, the slide here represents a gene, and these icons represent certain regions in the gene. So for example, if a variant were to fall outside the gene, such as the 5' to 3' region, so either here or here, excuse me, if it falls in the 5' or 3' region, the variant is unlikely to affect the gene itself, but likely affects the regulation. So for example, mRNA transcription and transcription activity in general. If a variant were to fall within the gene, so the yellow part. So, sorry, not the yellow part. Oops, apologies. So within this region, if the variant were to fall within that region, sorry, if the variant were to fall within that region, so for example, within the coding sequence, or the variant is more likely to affect the gene itself. If the variant were to fall in an intronic region, the variant would be unlikely to affect the gene, but might affect the mRNA stability. So, for example, splicing or translocation efficiency. If a variant were to fall within the coding region of the gene, so in the yellow parts here, these can have, as I mentioned earlier, these can have an effect on the gene in two ways. So it can have a synonymous or a non-synonymous effect. For synonymous variations, the protein sequence remains the same, so there is no change in the amino acid. But for non-synonymous, or also called missense variations, there is a change in the amino acid, and therefore the protein sequence. So we can say that it is likely that this variant would have a greater consequence to the gene itself and the function of the protein. So again, just to summarize, in Ensembl, we plot each variant to the genome, and depending on where the variant falls within the gene, we assign a consequence to the variant. And these variant consequences have special terms. So we also call these sequence ontology, or SO, similar to gene ontology that Alina mentioned earlier. It is a standardized way of describing sequence-based features. So each SO term, or sequence ontology term, is color-coded, and you can read more about sequence ontology in general and the different color codes in this link below here. And you won't have to memorize these colors. They will always be available in the legend on Ensembl. So when we talk about non-synonymous or missense variants, we rank them by their pathogenicity. There are different algorithms that rank these changes in the amino acid sequence based on how conserved the protein is. Also the chemical change in the amino acid position and the 3D structure of the protein and its domains. But please note that these are predictions and not facts. So it's always good practice to validate your prediction experimentally. So if you have a variant that you've identified bioinformatically, it's always good to do an actual experiment in the lab to verify this. Ensembl uses a number of different pathogenicity tools which rank these missense variants according to a scoring system. So for example, the first one here, the SIFT score ranges from 0 to 1, with 0 predicting a deleterious effect, and 1 predicting a non-deleterious effect. For for polyphen 2, the scoring system is inverted. So a 1 indicates a damaging effect, and a 0 indicates a benign effect of the variant. Then we also have the CAD score, which ranges from 0 to 100, with 0 indicating a likely benign effect. We have a few more of these pathogenicity scores. So for Revel, Meta-LR, and Mutation Assessor, they all have a scoring system from 0 to 1, with 1 indicating, the red here, the 1 indicating a damaging effect of the variant. And finally, I want to talk about reference alleles in variation data. So if you remember from earlier today, a genome assembly is made up of chunks of sequences from individuals, like a jigsaw puzzle. So when you take a sequence from the human genome, sorry, excuse me, when you take a sequence from the human genome assembly and look at a particular variant, for example, this T here in the sequence, it does not necessarily mean that it's a representation of all the human individuals around the world. So this T allele is found in this particular person, but it does not mean that the majority of people actually have this T allele. She might actually be a person who carries an allele that could cause susceptibility to a disease. So when we talk about reference allele, it is not always the major allele, the allele found in the majority of people, but rather it's the allele found in the particular genome reference assembly you're using. But most of the time, the reference allele is the major allele. Oh, sorry. All right, so let's move on to the demonstration. We are going to look at a gene called MCM6, and we're going to find variants in the gene, and we're also going to look at the region, so the flanking region of this gene. And then finally, we will look at a particular variant, so this one here, to find out more information about it. I'm going to copy the gene name and the variant ID. And I just pasted that in the chat box. So if you'd like to follow along, you can do that, but I'll also just go through it on my own screen. Okay, so for this, we will open a new tab and we'll go to ensembl.org. I'm just going to zoom in a little bit. Okay, so for the first part of the demonstration, I'll show you how to find variants in a gene. So the gene we're interested in is in human. So I would click this drop-down menu in the middle of the page. There is a search section. So I selected human here. Then I will paste MCM6 in the search bar below and then click go. Okay, so we're interested in MCM6 in human, so that's the first hit here. I will click that link, and that brings us to the gene tab. So you can find out which tab you're in by looking here at the top of the page. So here it says gene MCM6. So you've explored this a little bit with Alina, but what we're interested in are variation data. So you can find variation options here on the left-hand side. And first, I will show you the variant table. So I will click variant table, and that opens that opens the variant table on the right-hand side. So if you scroll down, you can see a list of all the different variants that have been identified in this gene. So the headers look a bit messy at the moment. That's because I am zoomed in. But if you zoom out, you can actually see that it'll adjust. But for the demonstration, I will stay zoomed in so you can see it a little better. Okay, so let's look at the different columns. So the first column, we have the variant ID, we have the chromosome location, and then also the allele. So the first allele is always the reference allele, and the second allele is the alternative allele. Then we've got global global major frequency, I think it is. Second most common. Yes. So if you are unsure.
Minor allelic frequency.
Oh, minor, sorry. Thank you very much.
You're welcome.
All right. Yeah. So if you're unsure, like me, you can hover over this, the headers that have dotted underlines, and it just gives you a little explanation of what each header means. And then we also have the class, so the type of variant. So in this case, it's the SNP. Then we've got the source where this variant comes from. Then also some evidence. So here we have evidence that it's from frequency, but also from the GNOMAD project. And then we've got the resulting amino acid change. So here it's glutamic acid to glycine. Then we've got some more statistical information. So here, oh, sorry, before the statistical information, we also have the amino acid coordinates. So the last part of the table has the consequence scores, that the pathogenicity scores that I've covered earlier. So if you just hover over each of them, you can see what these scores mean. But you can, if you look at the numbers themselves, you can also tell that they don't coincide with one another. So this is why it's always good practice to validate your predictions experimentally. And then finally, the last column, we have the transcript ID. So the transcript in which this particular variant was found in. So that's the variant table. Now I will show you how to view variants in the location tab. So I've scrolled up to the top of the page, and I'm going to click location tab here on the top left-hand side. And this opens the region and detail view, which you have explored with Alina. So we've got the chromosome view here. Then we've got the 1 megabyte view here. And then further down the page, which is still loading, that's the region and detail view. And in this view, you can add different tracks. So we can, for example, if you're interested in variants, which we are here, we can add tracks to this view to see what kind of variants are present in this region. So to do that, we'll click the configure this image button here on the top left, or it's the same as configure this page. So I'll click configure this page on the left, which opens a pop-up window. And here we can enable the different tracks. So we are interested in variation. So here on the left-hand side, we have a section called variation, and I would click click that link that says variation. And here we have different tracks that we can add to. For example, if you are interested in UniProt short variants, you can click this empty box, which opens a pop-up window, and you can select how you want to view it. So by default, at the moment, it's turned off. But if you're interested in that particular track, you can just click on one of these options to show them on the region and detail view. Let's say we are interested in 1000 genomes data. So here I would click, I want to see all variants of the 1000 genomes project. So I'll click the first one and just click normal. And then I'll also click the second one. So I'm selecting short variants and common variants. So the common variants, if we are unsure about some information, you can click this eye or this information icon on the right-hand side. And here it just explains what this means. So in this case, the common variants are variants that have been found in the population at least 1% of the population. And then you can close it again by clicking the eye icon. So here we have some other tracks already turned on. So EAS stands for the East Asian population. But if you want, you can turn these off. And activate or turn other tracks on that you're more interested in. So here, for example, I've selected the okay, I think I just found a little bug. So here in the description it says that it's the population of East Asian individuals, but actually SAS stands for South Asian. So I'm gonna report that to our team. And you can see that their ensemble is not just Alina and me. It's a big team made up of 100 people who work on this project. So we're always trying to improve things here. Okay, so I've activated the South Asian population tracks. And if you're happy with your selections, you can save these by clicking the check icon on the top right-hand side. And this updates the region and detail view. So this might take a little time because it's quite a big, it's quite a detailed image. Okay, so if you scroll down to the bottom of this view, you will see that we've got new tracks added. So here we've got the 1000 genomes tracks, specifically the SNPs and indels, and then also the common SNPs and indels. Then we also have the tracks for the South Asian population. So again, SNPs and indels, and then common SNPs and indels. We also have a track by default, it's turned on, which is called all phenotype associations. If you're not sure what a track represents, you can click on them, and then you'll find more information on these pop-up windows as well. So this view is interactive, so you can click the different blocks. So the colors, they represent different, they have different meanings. So if you scroll to the bottom here, to the variant legend, you can see the different color codings, so what they mean. So I'm just going to click on this, on one of these SNPs. And here, again, you have a pop-up, but it'll show you more information about the particular SNP that you've just selected. So we've got the ID, we've got the type, so whether it's a SNP or it's an indel, the location, the alleles, and then also the consequence type. So here, this one describes an intronic variant. Okay, so this is how you can view variants on the location tab. You can also view a particular variant. So we've looked at variants within a gene. But let's look at a particular variant now. So we've got the RSID copied from earlier, and I'm just going to paste that into the search box at the top right-hand side of the page. And then click enter. Okay, so by default, you land on a variant tab. So we haven't explored this one before. And this tab just shows you more information about the variant. So you will land on the Explore this variant display. And here you'll just see all the different details. So the consequence, the alleles. So this starts with the reference allele, and then for this case we have 2 alternative alleles. We've also got some evidence information, so the different projects or papers that it was listed in, whether it has any clinical significance, and then also some more information about the source and also their synonyms, its synonyms. Then further down, you have different display options. So all these views are the same views as the ones on the left here on the top left. So I'll show you the genes and regulation display first. So you can click on here in the middle of the page or here on the top left. And this table just shows you all the predicted consequences of this particular variant that we have searched for. So a single variant can have more than one predicted consequence because a variant can be located on more than one particular gene transcript. So if you scroll down, you will see that you'll have information about gene expression as well. So here we have the gene ID, and then just some summary statistics about the expression correlations. And then if available, it'll also show you if the variant falls within a regulatory feature or a motif. Okay, so that's the genes and regulation display. We also have population genetics information on this variant. So if you click population genetics on the top left here, it will open this window, which gives you all the population genetics information from the different projects that we take our data from. So the top of the page remains the same, so it's all the information about the variant. But if you scroll down, you'll see that the page is divided into different projects. So here we've got information about the 1000 Genomes project. Scrolling further down, you'll also see information, population genetics information from the NOMAD project, NCBI Alpha, TopMed, UK10K, and the Gambian Genome Variation Project. So we are going to focus on the 1000 Genomes project, so that's further up the page. And here you'll see a nice little summary of the allele frequencies within this project population. So we have a pie chart here, which just shows the frequency of each of the entire population, but also frequencies of the subpopulations. So African, if you hover over these names, African, American, East Asian, European, South Asian. Oh, that's right here. Interesting. But you can also look at the subpopulations of these subpopulations. So if you click the link underneath the subpopulation, you'll see that the African population also has a number of different subpopulations. And you can change these depending on which population you're interested in. So scrolling down, you'll see a table of the details of the allele frequencies. So they just give us a little more detail on each population in a table format. So we've got the population name. So again, you can hover over them, show you what population it is. Then we've got the graphical representation of the frequency. We've got the allele frequency count. So this is the number of total alleles. As we humans are diploid organisms, we have 2 copies of each gene, and therefore 2 alleles of a certain variant. So each individual that was sequenced in this project provided 2 counts to the allele frequency. And then we also have the genotype column, so the observed genotype of the different individuals in the population. And then also you can look at these in more detail on this link on the right-hand side of the table. If you click on that, so I'll click on the first line under genotypes. It will lead you to the bottom of the page, which will just give you a little more information of the observed genotypes in each individual. So right now it's loading. Oh, that was quick. So this is just a table that shows the genotype of each individual. So here we've got the sample name, the gender, genotype, and then the populations that they belong to. I'm going to scroll back to the top of the page, or you can also click this back to top button on the right, which is not working. Okay, I've just scrolled up to the page. So we've explored the genes and regulation view. We've explored the population genetics view. Now I want to show you the phenotype data view as well. So if you click phenotype data, it will open a table of all the different phenotypes that are associated with this particular variant. So scrolling down, you'll see what phenotype, disease, or trait it's associated with, the source. We've got some more source informations, and also a link to the publication if there is one available. You can click on the different phenotypes. So, for example, let's click on body mass index. And this opens a new page, a new tab actually, that is called phenotype here on the top left. So here we're going to see a little more details on this particular phenotype of this variant. If you have a phenotype that you're interested in, you can also type them into the search box. So let's say if you're interested in Alzheimer's disease, you can type it into the top right-hand search bar here, and that will just give you a list of results of all the different genes or variants that are associated with Alzheimer's. So if the Ensembl browser is a bit slow for you, which it is for me right now, we can also visit the mirror sites. So I'm going to, if you want to use one of our mirror sites, I'm going to click the Help and Docs link on the top of the page. And here on the left-hand side, you can search for mirror sites. And this just shows you, gives you the links to the other, the mirrors that we have available. So it might be a bit faster if you go to one of the other pages. I'm just going to go back to my first tab to see if it's loaded, and it has. But at least you'll know that you can use the mirror site now that if the browser is being a bit slow for you. And you can make sure that you are indeed on the mirror site by looking at this little icon here. So we are on the American US West mirror. I'll close these tabs because I don't need them anymore. And I'll just go back to my first tab. Okay, so here we have a table of all the associated loci that are associated with this phenotype, so body mass index. Just like the other tables, you can filter and sort these. So for example, you can click on these filters up here and select filters that you want. You can also change filters, but also, most importantly, you can download the entire table. So there is this Excel icon on the right-hand side. And if you click that, the browser will prepare the page for you to be downloaded and looked at on Excel. I'm actually going to stop that because I just realized it's quite a long table and this might take some time. But anyway, this brings me to the end of the presentation. So I'll go back to the slides and I'll let you have a go at the exercises in the training page. So just to remind you, I'll open a new tab and go to training.ensembl.org. And then you can find training materials here on the top of the page. There is a link. We are the first one. And here at the bottom, you can see the different demos and exercises that we have. So we are going to do the variation exercises now. So I'll give you 15 minutes to do that, and then we'll come back and I'll talk about the variant effect predictor. All right, so it's 11:45 now, and if there aren't any questions, I'll move on to the next module, which is looking at the variant effect predictor, or VEP. All right. So the variant effect predictor, or VEP, is a tool that you can use to annotate and predict the functional consequences of your own variants. So let's say you just received your sequencing data and you've called variants from this data, and now you want to find out more information. For example, the consequences of the different variants you found. And this is what you can do with VEP. So you can find out the effect of your SNPs, insertions, deletions, CNVs, and other structural variants. And this is how you use the variant effect predictor. So first, you input your data in one of these different formats. So I will talk more about that during the demonstration, because I think it's easier to just show you what the different formats look like. Then your data is processed by VEP and searches Ensembl and external databases to give you information, for example, the effect of the variant, the pathogenicity score, allele frequencies, and consequences, and also a list of publications if there are any available. And there are 3 ways to use VEP. So you can use the web interface on the Ensembl browser, which we are going to focus on. But you can also access VEP through the command line or REST API. So this was a very short presentation. I mainly want to show you how to use it, because I think the best way to get to know VEP is to actually explore it yourself and do a query yourself. So we have, for our demonstration, we have 5 variants on the human chromosome 9, which is a list, this list here. And we will use VEP to find out if the variants have already been annotated in Ensembl, what genes are affected, and also if the variants affect gene regulation. So I've formatted these variants. So I'm going to copy them and put them in the, oops. Put them in the, here. I'll put them in the chat box now. And you can copy this from the chat box and follow along on your own screen, or just watch me do it on my screen. But for this, we're going to go to Ensembl. So I still have a tab open. So this is still from our last demonstration, but you don't have to go to the homepage. You can just click VEP on the top, on the navigation bar here on the top of the page. So if you click VEP, this leads you to the different interfaces that you can use it with, but we are interested in the web interface, so I will click Launch VEP here on the left-hand side. Okay, so by default, you have Homo sapiens selected, but you can, if you are interested in a different species, you can also delete it by clicking this cross here and just change species by clicking the link below. So we are interested in the human chromosome 9, so I'll keep the species. So here is where you add all your information. So you can add a name for this job. So I'll just call it VEP demo. You don't have to add it though; it's optional. Um, and then in the next box, you can paste your data. So I talked about the different, I briefly mentioned the different formats that you can paste your data in. But what I actually want to show you is I want to show you how these formats look like. So here below this box, you have links for different examples. So if you click the different links, it just gives you an example, some example data in the input box. So this is the Ensembl default format, but you can also paste VCF formats, variant identifiers, so the RSIDs, HGVS notations, or speedy format. You can delete these because we are going to paste our own data. So I've got that copied and I will paste them in the box. You don't actually need to delete the spaces in between, but I just like how it looks without the spaces. So you might have noticed that this format is the Ensembl default format. So we have the chromosome in the first column, followed by the start position, end position, the alleles, the strand, on which strand it is on, sorry. So this is the plus stands for forward strand, minus for reverse strand. And also I've added some names for the different variants. So I just called them var 1, 2, 3, 4, 5. If you have a very big file, you can also choose to upload it instead of pasting it into this box. So
start giving us tag now and let's put it on this thing. Start giving tag so that once you see that tag, how many times will somebody be stopping on the road?
hi, sorry, can I ask you to, oh, okay. Do you have a question? Sorry. Okay, cool. I think that was just an accident. So where was I? Ah, yes. So we've just pasted the data here in the box, but you can also upload your file if you like. So these have to be in the same formats as the ones here in the example. Also, if you have a URL to the file, you can, instead of pasting it or uploading it, you can just provide a web with the URL. Next, we have transcript database to use. So we are interested in Ensembl GenCode transcripts, but you can also choose other databases. So RefSeq from the NCBI, or you can also choose to show both Ensembl and RefSeq transcripts. Then we've got the section called additional configurations. And here you can just add some more configurations if you like. So if we click on this first option here, identifiers, we can choose to show different IDs in our results. So you can choose to show the gene symbol, transcript version, but also if you're interested in the protein name or UniProt IDs. I will keep the gene symbol and the transcript version, and I will click identifiers again to minimize the window. The next configuration we have is variance and frequency data. And here you can choose to show whether your input variants are known or novel. So whether you want to find out if your input variants are novel or known. So this is turned on by default, but if you like, you can also change this option to no, or yes, but don't compare alleles. Then you can also show variant synonyms, so other names for the variant that you've inputted. And I can also see here, by default, the PubMed IDs for citations is turned on. So here you will get a list of a link to the available publications that have cited this, your variant. So I'll close this and go to the next one, additional annotations. And here you can turn on different annotations, for example, the transcript name, or if it falls within a regulatory region, or any associated phenotypes. So you can look at the different options here. I'll turn on the phenotypes option because I'm interested in it. But next, if we close this and go to the predictions. So here we have those pathogenicity scores that I briefly mentioned in the variation module. So you can choose to show the different scores that we have available for you. So by default, we've got the SIFT and polyphen scores turned on. I close the predictions now. And then finally, what I want to show you is the filtering options. So if you like, if you're very specific about your research, you can apply filters before you even get the results. But it's always good practice to apply filters after the job rather than before, because this way you won't lose any potentially useful data. So by default, this is turned off, and I will also keep it turned off. Then, once you're happy with your settings, so you're happy with human, we've given it a name, our data, our database, and the different configurations. Once you're happy, you can click Run. So on this green button on the bottom of the page. And I got an internal server error. So I would just refresh the whole page and input our data again and try again. So we have the name, we've got the variants, we've got the database. I'm happy with the identifiers. I want to see known whether the variant is known. I don't think I changed anything here. We have some predictions, okay. And the filtering option is turned off. Okay, let's click Run again. And this will show you, it will send you to the page where you have all your recent jobs that you have submitted. And this page gets refreshed every 10 seconds, so you can see it here, or you can refresh it manually as well. But once it's ready, so at the moment you can see it's still running, but once it's ready, it'll have a green button that says Done on it. Oh, like here. But before we see the results, I just want to show you, you can also save this web query on your account. So you can register on Ensembl here on the top right-hand side of the page. If you have a lot of data that you want to keep track of, so you can save the different queries that you're making. It's free, and you can ask to remove your profile at any time. So you can save this to your account. You can also edit it if you realize that you wanted to make a change in your query. But you can also share this particular job. So if you have collaborators that you want to show your results to, you can click this button and just copy the URL and send it to them. Or you can delete it, but I'm not going to do that. I'm going to look at the actual results now. So you can click on Done. And this shows you all the results from our 5 variants that we've inputted. So at the top of the page, we have some summary statistics. So we've got the number of variants that were processed, so it were 5. Then we've also got the number of variants, so the number of these 5 variants, whether they are novel or existing. So we can see here that 1 variant of the 5 was novel or is novel, and 4 of them have already been cited before. Then we've got the number of overlapping genes, which is 4. We've got 24, it's overlapping 24 transcripts. So remember, 1 gene can make up not just 1 transcript, but multiple transcripts. So that's why we have a higher number in the transcript section here than in the gene section. And then we can also see that our variants have overlapped 2 regulatory features. Then on the right-hand side, we have a little pie chart that just shows the consequences of the variants that we identified or that we've given. So we have the type of consequence, so missense, for example. So in 32% of the results, this variant is missense variant. And then also we've got the coding consequence here at the bottom. Now, if you scroll down, you'll see that you have this table. By default, the table is not or might not be sorted properties. So what I like to do is I just like to click this little arrow to make sure that the table is sorted according to the variant names. So we have variant 1, followed by variant 2, and so on. Now you can also see that there are multiple entries for each variant. So this is because there can be multiple consequences on multiple transcripts of a gene. So looking at the table, we've got the name that we've given, we've got the location of the variant, the allele, the consequence of each of the results, then the gene symbol, also the gene ID, Ensembl gene ID, sorry, and also the transcript ID. Then we've got another column that describes what this particular sequence is. So this is a protein coding sequence. And then we've also got some more location information. And if it's available, so if this variant is not novel, so it's been cited before, you will be able to click these links that will send you to more information of the known variant. Scrolling further along the table, you'll also see that we have all these pathogenicity scores that we've turned on. So we've got SIFT and polyphen, and then also the clinical significance of the variants. So in this case, the majority, or maybe probably all of them, are benign. But what I want to show you mainly is this link here. So under the PubMed column, if there is a link available, this means that there is a publication out there that you can go to and read and find out more information on what the researchers have found about this variant. So I'm going to scroll to the start of the table again, and I want to show you the filter options that we have. So above the table, you see this filter box. And if you're interested, for example, in a particular consequence only, you can apply a filter. So you can click this drop-down menu and select the column that you want to filter. So I want to filter the consequence column. And then, so we are interested in let's say we are interested in missense variants only. So I will type in missense variant, or it'll find the different consequences. If you are not interested in the missense variant, so if you want to show everything but the missense variant, you can also choose the different options here, so is not, and so on. And make sure you click on Add to apply your filter. And that just updates the table here at the bottom of the page. So this might take a little time if you have quite a lot of results. Our table is okay, so it shouldn't take too long. Let me try a mirror. Oops. Oh, okay. I was just about to start to do another query on a mirror, but it just loaded. So now you can see that the table has been filtered to show missense variants only. And you can also apply multiple filters. So let's say maybe you're interested in main select transcripts only. So for this case, there are only two options, whether it is a main select transcript or not. So here you can choose is or is not, and we are interested in is. So we'll click Add, which updates the table again. So now we have, in our results, we now have variants that are main select located within main select transcripts and are missense variants. You can also delete your filters if you've made a mistake or if you want to change it by clicking this cross icon on the right. And then finally, you can also download this data. So you can, under this download box here, you can select the format that you want, so VCF, web, or text format. And if you click on one of them, it just opens your download system viewer. I'm not going to save that though. Okay, so this is the variant. This is the variant effect predictor. We are halfway through, so I think we'll give the exercises some time. So let's say 10 minutes, 15 minutes, and then afterwards we'll take a lunch break. So let's try the web exercises for 50 minutes for now. Okay, it's 1:35 now, so I'll get started. Excuse me. Sorry, my throat is a bit dry, so if you hear me cough, I apologize in advance. Um, okay, so welcome back, everyone. I hope you had some time to have some lunch and also just to walk away from the screen for a little bit. We're going to move on with the rest of the workshop. So this afternoon, I'll be talking about comparative genomics, regulation data on Ensembl, and also BioMart, which is a tool that you can use on Ensembl to extract large amounts of data. So let's talk about the comparative genomics data available on Ensembl. We are going to look at two main themes in this module. So I will talk about the applications of comparative genomics and the species that are available on Ensembl. And I will introduce you to the comparative genomics data we provide at the gene level. So, for example, gene trees and homology predictions. And then in the second part, I will talk about whole genome alignments and shared synteny. Let's look at the applications of comparative genomics first. So researchers in this field are mainly interested in examining the evolutionary relationships between species, find and understand differences between genomes, derive a gene's function based on homology, and identify highly conserved regions within the genome. There are many different ongoing projects that generate sequencing data for researchers to do the comparative genomics analyses on. So remember, Ensembl does not do any of its own sequencing, but rather we retrieve publicly available data and apply our annotations to them. On Ensembl, we perform comparative genomic analyses by taxa. So this means that we compare all vertebrates against one another, all plants against one another, the metazoa against one another, and so on. We do not perform these analyses on bacteria, though, because there are just too many of them. There are too many available, too many genomes available. You can view this data under the Gene tab, which I will show you during the demonstration. But just as an overview, you can see it highlighted here. On Ensembl, we also perform pan-taxonomic comparative analyses, so comparative analysis across different taxa. For this, we take a number of representative species from each taxa, so some from protists, some from plants, some from fungi, some from the vertebrates database, and a few bacteria. Similarly to the within taxon comparative analyses, you can find the views of pan-taxonomic data under the Gene tab. So here we have the within taxa analyses, and here we have the options to view the pan-taxonomic analyses. We also offer comparative analysis data within species. For example, we have mouse data, which includes all mouse strains and species available on Ensembl, but we also do this for rat, and we have similar comparative analysis data available for pig breeds and other agricultural animals. Sorry. A type of comparative analysis data we produce are gene trees. So for this, we take a representative protein of each gene from each species. And that's why we can call these trees protein trees as well. And we classify them, we classify the representative proteins into families or using different other clustering methods. Then we align these, reconcile these against species trees, and that's how we produce the gene trees. And then finally, from these gene trees, we can make homolog predictions. So there are two main types of homologous relationships, orthologs and paralogs. So orthologs are genes that have emerged through a speciation event, which is highlighted in blue here on this gene tree on the right. So for example, C1 and H1 are orthologs, sorry. They have an orthologous relationship because they came from a common ancestor, from a common gene, genetic gene that split these two species have genes, and they have an orthologous relationship because they've emerged the genes have emerged through a speciation event. So orthologs can also be described as one-to-one or one-to-many, which you can see here. So C1 and H1 are one-to-one orthologs because they are only orthologs to one another. And we also have the one-to-many orthologs, which would be M, so the mouse gene here, to C1, H1, C2, and H2. So the mouse gene is related to all the other genes by an orthologous relationship because they've emerged from through a speciation event, which you can see in this node here in blue. The second type of homologous relationships, the second type of homologous relationships are paralogs, and these are genes that have emerged through duplication events. So represented in red here on the tree. So for example, C1, and C2 are paralogs or have are paralogs to one another, and H1 and H2 are also paralogs to one another. Orthologs often keep their function in the course of evolution, whereas paralogs may evolve new functions. Okay, so in our first demonstration of this module, we are going to look at the BRCA2 gene, and I'll show you how to find the gene trees and also its homologs. So I will copy the gene name and paste that in the chat box. And I will open a new tab and go to ensembl.org. Okay, so we want to look at the BRCA2 gene. So I will paste the gene name here in the middle of in the search box in the middle of the page. And then I'll click go. And I'd like to look at the human gene, so I will click this first hit in our results. And that leads us to the gene tab. So you can see which tab you're in by looking at the top of the page here. Excuse me. Okay, so I've shown you this little overview at the beginning of the presentation, but here you can find the within taxon, comparative analyses, and then I'll show you the pan-taxonomic analyses, where to find them later. Okay, so let's look at the gene tree. So I will go to the left-hand side here and click on gene tree. And that just returns a page with the tree, as some summary statistics, and also a legend at the bottom. So let's go to the summary statistics first. So gene trees have their own Ensembl ID. So Ensembl IDs always start with ENS, and then GT stands for gene tree, and then a number, a generated number. And this particular gene tree has 176 genes. It's got 168 speciation nodes, three of which were 3 duplication nodes, and 3 ambiguous nodes. If you scroll further down, you can see that the tree is divided up into different clades. So our gene of interest, or our species of interest, is highlighted in red, so BRCA2 in human. And then we've got the closest relatives. So we've got gorilla, gibbon, and so on. And then we've got in orange here more primates that are a bit more distantly related to us. Then we've got some other mammals as well, frogs and bony fishes. So the clades are colored in different blocks, have different backgrounds, colored black backgrounds. And you can also see these triangles. So if you click on them, it'll describe what they represent. But basically, these triangles are collapsed subtrees. So if we were to expand the entire tree, it would be very big, and it would be very difficult to keep an overview of it. So a lot of the subtrees have been collapsed in these gray funnels. So if you click one of the gray triangles, which I've done here, you can choose to expand that particular tree you clicked on. You can expand all trees. You can also collapse other nodes, and you can export the tree or even the sequence only. So if you click on expand this tree, it will just load the gene tree image again. And now you can see that here we've expanded this particular gene tree, subtree, sorry. So if you are not sure what the different colored lines or nodes mean, you can go to the legend at the bottom of the page, and you can just see here, for example, the nodes. The dark blue nodes here are speciation events. Red nodes would dictate duplication events, and so on. On this particular tree, it doesn't seem, we cannot see a duplication node, so I assume it's within one of those collapsed subtrees. And then here again, we also have the gene ID of interest and gene ID the homologs. The legend might look a bit different on your screen, and that's just because I'm zoomed in, so the text has overlapped. Okay, so this is the gene tree, but to the right of it, we also have a graphical representation of the protein alignments that we use to build this tree. So the dark green lines, or the dark green alignments, represent a consensus alignment of the collapsed tree that it is on. So basically, this alignment here is a summary of all of the mammals within this subtree here. And the light green alignments are alignments of the individual species. So if you go to the bottom of the page here, you can see that the white color represents gaps in the alignment, and the light green color represents alignments. So we can see here these green blocks, that's where sequence is aligned, whereas here they did not align. So this is the gene tree. What I also want to show you are the orthologs. So if you scroll back up to the top of the page, you can click orthologs here on the left-hand side. And this would just open, I think it's, this would just open a table of all the orthologs of our gene of interest, so BRCA2. Okay, so if you scroll down, we can see that the first thing we have is a table. So this is a table of all the different species sets that we have. We've got primates, we've got rodents and related species. I'm not even going to pronounce that. Carnivores, ungulates, and insectivores. Placental mammals, sauropsida, fish, and then also by default, you've got all species selected. So you can choose to view just a certain set of species by clicking on this box. But by default, you will be able to see all of the species. And if you scroll further down, that's where you see the list of orthologs of all of these species. And before I move on, I just want to show you some of the numbers that we have as well in the table. So we've got the number of one-to-one orthologs, we've got the number of one-to-many orthologs, many-to-many, and then also the number of species without orthologs. Okay, so I'm actually going to filter this table. I just want to look at rodents. So I'm going to select rodents from the first table. And that automatically refreshed the bottom table. And now if we scroll down, we can see that everything we see is just rodents and related species. So we've got the species name in the first column, then the type of ortholog, so what it is. In this case, it's a one-to-one ortholog. The gene within which the name of the gene this ortholog is of. So in the Algerian mouse, in this case it's BRCA2 as well. You can compare the regions, so the human BRCA2 and the Algerian mouse BRCA2 by clicking compare regions. And you can also view sequence alignments. So I'll do that in a minute, but I just want to show you all the other columns as well. So we've got the target ID. So that's the ID that matches, this is very complicated. So this is the percentage that matches the human sequence of the orthologous sequence. And then we've got the query ID. So this is the percentage of the human sequence that matches the ortholog. Then we've got some gene order conservation scores and some whole genome alignment scores as well, coverage score as well. So we can view the sequence alignment of human and Algerian mouse BRCA2 by clicking this View Sequence Alignment link. And then you can choose to view either the protein alignment or the cDNA alignment. I want to look at the protein alignment, so I will click the first link. And this opens a new view. So this is our ortholog alignment view. We've got the two species that we are looking at right now. So human and Algerian mouse, just some details on this ortholog. So the gene ID in both species, and also the length of the alignments. And then if you scroll further down, here is where you can see the actual alignment of the protein sequence. So in this case, it's in the cluster W format, which is one of the many multiple sequence alignment formats out there. You can, if you are unsure about what you are seeing in a particular page, you can always click on this question mark icon whenever you see it. And then also another important thing, if you want to download the sequence alignment, you can click on download homology. So I will click this button now. And this opens a pop-up window where you can select the different of the file format that you want to download. So we've got little previews here as well. So the one we just saw is the cluster W format, but you can also download it in the FASTA format, Mega, and so on. So here it just gives you a little overview in case you've forgotten what they look like, because there are a lot of different formats out there. You can change your name here. You can also select the format from this drop-down page. And then once you're happy to download, just click the download button. Or if it's quite a big file, I suggest you download the compressed version of it. Okay, so I will close this because I don't want to download anything for now. And that brings me to the end of our first demonstration. So we've looked at how to find the gene tree information and also ortholog information of a certain gene. I'll go back to the presentation and move on with the next part of our module. So in the first module, we looked at the gene-based comparative genomics data available. So like I said, gene trees and homology information. But we also have whole genome alignments available. So these are useful to identify highly conserved regions in the genome or sequences that have evolved slowly and are likely functional. And they can include both coding and non-coding sequences. Whole genome alignments are also useful to support problematic gene predictions and define syntenic regions, which I'm going to talk about later. And on Ensembl, we have two types of whole genome alignments, and these are pairwise and multiple. In sorry, pairwise alignments are performed with BLAST-Z, or the most recent version, LAST-Z. And so BLAST stands for Basic Local Alignment Search Tool, I think. But it's an algorithm that allows you to align sequences. So pairwise alignments are performed in pairs, as the name suggests. So two genomes are aligned against one another. On Ensembl, we align human against all the other species that we have available. We also align model organisms against related species, and also some agricultural mammals. We also perform multiple alignments against predefined species sets. So in these alignments, instead of aligning one genome against one another, like in the pairwise alignment, in multiple sequence alignments, we align multiple genomes against one another. So there are different multiple alignment analyses and species sets that we have. So these are named all here. But for example, in our EPO analysis pipeline, we have species sets of fish, sauropsids, eutherian mammals, and primates. So within each set, we would perform multiple sequence alignments of all the species. So we would perform multiple sequence alignments of all the primates, and then multiple sequence alignment of the eutherian mammals, and so on and so on. If you want to find out the list of species, you can click on this link here in the presentation if you are interested. And then finally, I also want to mention synteny. So synteny is the conserved order of aligned homologous blocks between species. So let's say we performed a pairwise alignment of human and chimp, so human here, chimp down here. And once aligned, you'll notice that blocks of homologs keep their order. So you might not be able to read it here because it's quite small, but here we've got these four genes, ALG, UTP, NEC, and another NEC gene. And when we align these sequences against our chimp, we can see that the order is retained. So the order has been kept. So ALG, UTP, NEC, and the other NEC as well. And this is regardless of the direction of the strand. Okay, so in our second demonstration, we are going to look at this region in the human genome. And this region contains the HOXD cluster. So we want to look at the alignments and the conserved regions as well. So as usual, I'll copy the region in the chat box. Oops. Paste it in there. And I will jump out of my presentation and go back to the Ensembl tab that we opened earlier. So if you want to search for your location, you can either go back to the home page and search it from there. But what I'm going to do is I'm just going to use a shortcut because I'm lazy. I can paste the location here on the top right-hand side. And then what you can also do is, if you select this Ensembl icon here, if you click on that on the left, you can search human only. So we're going to search this region in the human genome only. Once you're done, you click search, which I've just done there, and that opens the location tab by default. Okay, so we've got our region and detail view. So you're familiar with this already. We've got our chromosome view, followed by our 1 megabyte view, followed by our super region and detail view. And we selected this region because we are interested in the HOXD cluster. So if you look in the middle of the 1 megabyte view, you can see that we've got all the HOXD transcripts here. So we can configure the region and detail view to give us more information about the comparative genomics data that we have available for this region. So to do that, you can go click on this cog icon to configure the image, or click on the cog icon on the top of the on the left-hand side of the page. It's the same one, but if you click that, you will it will open a pop-up window, and here, which we have looked at earlier, you can select different tracks that you want to see, so different annotations of your sequence of interest. So you explored genes and transcripts with Alina, then we've looked at variation, and now what we're interested in are comparative genomics tracks. So these are further down. I'll click the first link here on the left. And on the right, you'll be able to see all the different tracks that we've got available. So we've got these species sets, or these comparative analysis pipelines that we've got in Ensembl. You can click on the information icon on the right if you want to find out more. Then we've got conservation regions. So this will annotate the regions that are highly conserved within these species sets. Then we can also look at pairwise alignment data. So here we can select different species that we want to align against our human region. So what I'm going to do is, for the multiple whole genome alignment, I'll select the 91 Eutherian mammals. So this is a list of 91 mammals that we have predefined and done a multiple alignment to. And then also, I'm interested in some conservation scores. So what I'll do is I'll click this conservation score for 91 Eutherian mammals. So to activate a track, you just click this box on the right of your track of interest. And then further down, I also want to select some pairwise alignments. So let's just choose, I don't know, take cat. So I click the box. At the moment it's turned off, but I want to look at it in a normal view. Then I'll also choose a more related species, so chimp. Again, normal view. And then something further, more distantly related. Yeah, snake. Okay, so if you're happy with your selections, you can click this tick icon on the top of the right page, and that updates our region in detail view. Okay, so as you can see, you have to be a bit patient with Ensembl sometimes because um, in this case, we selected quite a few tracks, so it had to refresh the page and find this information. Okay, so here, so we have our chromosome, our region of interest here. We've got the conservation scores. So in this case, we've got a scoring system called GERP, which you can read more about here. But basically, you've got these two thresholds. So we've got 1.96 and negative 2.35. And what the numbers mean is the higher the number, the more conserved a region is. So you can see, for example, in the second track, the blocks represent regions that are conserved between the two, well, sorry, not between the two, between the 91 species in the species set. And then the gaps represent gaps. So sequences, regions that have not aligned against one another. And then you can see if you look, for example, in this region here, you can see this region is highly conserved within those 91 species. And you can also tell from the GERP score here that it is quite a high number. So that's our whole multiple genome alignments data that we've selected. So if you scroll further down, you can also view the pairwise alignments that we selected. Yeah. So here at the bottom, from earlier, if you remember, we've selected three species: cat, chimp, and a snake, tiger snake. And because we're most closely related to chimp, we have, if you look here, the colored box represents alignment, and the gap, for example, in the snake track, represents regions where the sequences have not aligned. So for chimp, we can see that this entire region has actually aligned. This makes sense because we have very similar genomes. Then if we look at the cat region, we can see that it's still quite aligned, but there are a few gaps in the sequences. And then if we look at the snake, the most distantly related species, we can see that it is quite a gappy region. Okay, so this is how you view multiple and pairwise sequence alignments in as tracks, but you can also view the actual alignments of a sequence. So if you scroll back up to the top of the page, which I've done here, you can go to the left-hand side. You have an option to look at the alignments. I will select the text. And this opens a new window on a new section on the right. So in this case, we will need to select our species of interest first that we want to align our region to. So you can click Select Alignment, and you can either choose if you want to do a multiple or pairwise alignment. So you've got these species sets here, and for pairwise, you have all the different individual species groups. If you know what you want to look at, if you know what species you want to look at, for example, mouse, you can just type it in at the top of the page and select it from this drop-down. And then to make sure that you indeed selected it, you can see on the right-hand side here that it's been added to our list. Okay, so I will click Apply to save my options here at the bottom of the page, which updates our view, and it gives us a table of the regions that aligned between these two species. So in this particular region, so this region, this HOXD region that we've selected earlier, we can see that there are two blocks of sequences that have aligned. So we've got one block, which is this length, and then the second box, which is much smaller. You can view the location on the human genome, but you can also view the location on ancestral sequences. So I'll click on this, the biggest, the longest alignment block. So that's block 1. Click this link. And if you scroll down to the bottom of the page, this is just a preview of our alignment. So you can click Display Full Alignment, and this just shows you the entire alignment. So now you can see that it is much bigger here. So we've got on the first line we've got Homo sapiens, human, and then on the second line we've got mouse, and then you can also see similarly to the sequence files that we've looked at earlier, the coding regions are highlighted in red. And yeah, you can just scroll down and look at it more. You can download this entire file by going to the top of the page and click Download Alignment. Or you can just view them on the browser. So here we can view it on human, but we can also view it on the mouse here. And then lastly, I also want to show you a region comparison, so a direct comparison, side-by-side comparison. So we've looked at the different tracks, the different whole genome alignment tracks that we can add. We looked at the actual alignment files, so the alignment sequence alignments, and now we can also view a region comparison. So this display is similar to the region in detail view. We've got our chromosome view, 1 megabyte view, and then region in detail view. So for this, we again have to select a species by clicking Select Species or Regions below the chromosome view. And this opens a pop-up again, and I will select mouse again just to show you how it looks. It's been added to the list, and when I'm happy, I'll click Apply at the bottom of the page. And now you can see that we've got our human region of interest,
the expression is higher when the person has an ulcer.
I'm guessing that was not a question. Just if you have a question, just put it down in the chat or in the living doc, or yeah, just ask away. Okay, so going back to this, we have our human region of interest. And then because we've added the mouse, we now also have our mouse region of interest. So you have a side-by-side comparison of the two species. And you can also look at them in the region in detail view. So here you can, like earlier, you can add different tracks and different annotations that you're interested in and compare them between the two species. Okay, so this brings me to the end of my demonstration. I will go back to the presentation and I will let you have a go at the exercises. So we are going to look at the comparative genomics exercises now. So we open a new tab, go to the training page, training.ensembl.org, and then click training materials here at the top of the page. It's the first link, Baze University. And we want to do the comparative genomics exercises. So I'll give you 15 minutes to do that, and then we will come back and continue with the next module, which is regulation. Okay, so it's 2:40 now. Before I move on to the next module, does anyone have any questions? All right, so let's move on then. So in this module, I'm going to talk about the regulatory, oh my goodness, I can't pronounce this word, regulatory data available in Ensembl. So I'll give you a brief overview, a brief introduction to the regulation of gene expression, and I'll talk about the Ensembl regulatory build. So how the regulatory data is generated and where this data comes from. On Ensembl, a number of genomes have been annotated with regulatory elements or features that control gene expression. For example, these are promoters, enhancers, and repressors. To annotate these regulatory elements, we look for specific biological data types. So for example, epigenetic marks, so histone modifications and DNA methylation, and transcription factor binding sites, and RNA polymerase patterns. And we also look at the open and closed chromatin conformations by looking at DNase 1 sensitivity. Essentially, each cell of an individual share the same genome and therefore the same genes. But we also know that there are hundreds of different cell types with very distinct phenotypes. And this is because of different expression levels of different genes. And that is why the regulation of gene expression is very important. So let's look at the different blood cell types, for example. They all have very distinct functions. For example, red blood cells are important in oxygen transport, or basophils, a type of white blood cell, are involved in allergic reactions. And all of these cell types are derived from a single hematopoietic progenitor cell. What makes blood cell types different is how they express genes at different levels and at different times. So we can say that these different cells have different epigenomes, or different epigenetic expression profiles. You can think of transcriptional regulation as layers of controlling the different timings and expressions of genes. So this is depicted here in this diagram. So we have a strand of DNA, and here we have, here at the bottom left, we have the coding region of our gene of interest. Upstream of that gene, you can find the promoter region, which binds RNA polymerase, the enzyme that transcribes DNA to mRNA. And the promoter region also binds transcription factor proteins. So these sites are also known as transcription factor binding motifs or binding sites. And transcription factors are involved in inhibiting or stimulating the transcription of a gene. Another regulatory element are enhancers. So these sites are often located a bit further away. So if we follow along the strand, we would find this here in this region. And these enhancer sites have their own set of transcription factor motifs, and they bind activator proteins which promote the activation of transcription in the promoter region. And then similarly, we also have repressors and insulators that bind specific proteins to downregulate gene expression. The binding of these transcription factor, the binding of proteins to the transcription factor binding motifs or sites is regulated by methylation. So within the DNA sequence, there are regions known as CpG islands, as you can see here. And these are regions that are rich in CG repeats. An enzyme called DNA methyltransferase moves along the genome and modifies the C bases within these CpG islands with methyl groups. So this gives different methylation patterns, and these methylation patterns can modulate the binding of transcription factors. So for example, methylation can block a transcription factor from binding to a transcription factor binding motif or site. So DNA methylation is one layer of transcriptional regulation, but there are many other layers of transcriptional regulation. So for example, the conformation of the chromatin can direct the activation or deactivation of gene expression. In this diagram, you can see that the chromatin is usually bound in a uniform manner. The DNA, so this is a zoomed in image, the DNA strand is wound around 4 histone proteins, and these structures are known as nucleosomes. So we know that there are regions of chromatin that are densely packed together, like here. Apologies. We have regions in the genome that are densely packed together, like here. And these, when they are densely packed together, they're often associated with transcriptional deactivation. So there isn't enough space for transcription factors to reach the transcription factor sites. And we also get regions of chromatin that are more loosely packed, like here. And this is associated with transcriptional activation because the DNA is more accessible to the transcription proteins. We also know that the position of the nucleosome and the modification of the histone tails themselves can affect transcriptional regulation. And then finally, lastly, the actual 3D conformation can also regulate gene expression. These layers of transcriptional regulation can be assessed by different experiments by looking at the expression patterns of a gene. So in this diagram, it just shows the different techniques that can be used to determine these layers of transcriptional regulation. For example, we can use DNA's sequencing to assess open or closed regions of chromatin by looking at the sensitivity of the DNase enzyme. We can use ChIP-seq to understand protein binding patterns and histone modifications. And we can also determine gene expression levels by looking at RNA-seq data. So now that I've given you a brief introduction to transcriptional regulation, I'll talk about the process of how Ensembl uses this kind of data to annotate regulatory elements in the genome. So first, we take these different types of data and import them from different sources. Second, we process the data to predict the positions of regulatory features, for example, promoters, enhancers, transcription factor binding motifs. In step 3, we assess the activity of these different regulatory features in different cell types. And then finally, we display this regulation data on the browser. Where does this data come from? So there are 3 main projects that Ensembl takes data from. One is the ENCODE project, which looked at 48 different human and 8 different mouse cell lines. They generated data that looked at histone modifications, transcription factor binding motifs, RNA polymerase activity, and DNA sensitivity. Then the roadmap epigenomics project looked at gene expression in cultured primary cells. And then finally, the blueprint epigenome project generated ChIP-seq and DNA sensitivity data of 20 primary hematopoietic cells. In Ensembl, we only have regulation data for human and mouse, but we are working on expanding this to other species in the future. So in a closer look in the first step of the Ensembl regulatory build, we plot the raw data against the genome using several algorithms and we annotate peaks. So you can see the raw data colored in these squiggly lines here, and then this blue straight line represents the reference genome. So for example, we start with data that assessed the activity of transcription factors and histone modifications. So here, transcription factors B and C and histone modifications 1, 2, and 3 display high activity in this particular region of the genome. When an activity hits a certain threshold, this peak gets annotated. So they are, we also say that they are all co-located in this region. And this is important to know for the next step of the process, where we move along the genome and find out if any other regions have that exact same pattern of activity of co-located regulatory elements. If any of those positions are, for example, known promoters, we infer that all the other positions with the same pattern or signature are also promoters. So we predict these feature positions. Then we look for other patterns, and then we do the same process again. Afterwards, we categorize the different regulatory features based on the underlying pattern or signature. So this process is called segmentation. And for example, this particular pattern, for example, is known to be associated with a promoter sequence, or this pattern here may be known to be associated with an enhancer. Next, we predict the cell type activity. So we do this for every cell line that we take from the different project sources that I mentioned earlier. We move along the genome in 200 base pair blocks and ask, is there a regulatory element annotated in this particular block? And if there is, we annotate that block. And we move on doing this along the entire genome. Oops. So this track here on top is what we call our multi-cell regulatory build, or the annotation of regulatory features for all cell types combined. Then we take this multi-cell regulatory build and we look at the individual cell types and we check the activity of the regulatory elements. So we ask, is this element active? Is it inactive? Is it poised or is it repressed? And to determine this, we look at the different experimental evidence that provided in the project sources. And then finally, in the last step, we display this information as tracks on Ensembl. I want to highlight that Ensembl does not link regulatory features to genes. We simply provide you with the locations of the features. So we also don't link regulatory features to gene expression, but rather we present cell line specific regulation data and tissue specific expression data. Therefore, when you are using Ensembl, we do not make inferences about the regulatory data, and it's up to you to make those your own inferences. Okay, so in this demo, we are going to look at the KPNA2 gene region, and we're going to find regulatory features in this region and explore its expression in different cell types. So I'm going to paste the gene name into the chat box, and I will have a sip of water first. Okay, so I'll jump out of my presentation and go back to our Ensembl tab, which we opened earlier. So we want to look at KPNA2 in human. What I'm going to do is I'm going to paste the gene name on the top right-hand search box and then hit enter. So it's KPNA2, human gene. This is what we're interested in. I will click the first hit in our results. And this leads us to our to the gene tab. But what we're actually going to look at is the location view of this gene. So I'll click the location tab on the left-hand side of the page, and this opens your very familiar region and detail view. So I will scroll straight down to the region and detail view, which is still loading. Okay, so we can see this view here. We still have some of the tracks that we turned on earlier, but if you were to reset this, you can, the configurations, if you wanted to see the default view, you can click on this cog reset button at the top. But for now, I'll just leave it because I know what we're looking at. So we're interested in the regulatory build track. So if you search along the different tracks, it should be further down at the bottom of the page. And you can see that here. You can also, these tracks are interactive. You can also change the order of the tracks by clicking the track of interest and then just dragging it along. So I've moved the regulatory build track at the very bottom now. Like before, the regulatory build track has its own legend, so we just search for it here, and you can see further down at the bottom. We know the dark red is promoter sequence, and the kind of lighter red is the promoter flanking region. So let's look at the promoter region of this gene. So I'll click the promoter box, so the dark red box. That will open a pop-up window, and it will give you more information about this particular feature. So we have the stable ID, the type, so we already know it's a promoter. We've got the location of this feature, and the things you learn. So you can also mark this feature on a page, on an image, or even zoom into it. But as it's already quite zoomed in, we don't have to do that. If you might want to do that here, where all the features are a bit closer together. So I'm interested in this particular feature. So to find out more information, we can click on the stable ID. And this opens a new tab called Regulation. And you might have noticed that this regulatory feature has its own Ensembl stable ID as well. So we have the ENS prefix followed by R, which stands for regulatory feature. Okay, so in this Regulation tab, you are able to see your regulatory feature in more detail. So here we've got our regulatory build track, which we clicked on just earlier. But then we also have other tracks. So for example, here we've got the Phantom 5 track, which is stands for Functional Annotation of Functional Genome. It's a project that annotated regulatory features in the human genome. Then we also have a tar-based track, which is another project which in this case, annotated microRNA targets. So we can see that along this promoter, we've got a few Phantom 5 annotations and some tar-based annotations as well. Then we also have this motif features track. These are the transcription factor binding sites or transcription factor binding motifs. The gray boxes represent the black boxes represent experimentally verified sites. So if you click on a black box here, you can see that we have the stable ID with the location, and then we also have some information such as the transcription factors that actually bind to the site, but also the different cell lines where it was experimentally verified in. The gray boxes, these are transcription factor sites that have not been experimentally verified. So these are predictions. So if you click here on a gray box, you can see under experimentally verified in, we have not available. Okay, I'll close this again and I'll scroll to the very bottom. Again, here you have the legend of the different color codings and also the legend of the motifs there. But at the bottom of the page, you have a list of the different cell lines where this regulatory feature, so this promoter, is active in. So we've got, we know that it's active in one cell line in astrocytes. So these are all verified. We know that it's inactive in this list. So if you scroll to the bottom, you can see your entire list of cell lines, then poised in 17 of the 118, and then repressed in 36. If you are interested in one particular cell line, you can look at it in more detail by going to the top of the page and clicking details by cell type. And here we can, we have this detailed view again, and we can add tracks to show the activity in different cell types that we might be interested in. And then also look at the corresponding evidence. So if you want to add a cell type, you can click on Configure Cell Tissue on this button above the image, which opens a new pop-up, and it leads us straight to the regulation section here on the left-hand side. So here we select, so first we select our tracks. So we select our cell type. Oh, sorry, this is, okay, I just realized that I've done a search earlier. So I'm gonna reset the configurations so that you can see what it looks like from the very beginning. So again, this has reset. We have only the default tracks turned on. So I'll click on Configure Cell Tissue again. And now, if you click on Select Tracks, you will land on the Cell or Tissue tab. So here, you can select your tissues of interest, your cells of interest, by going through the alphabet or just by typing it into this little search bar. It looks a bit awkward right now because I'm quite zoomed in, but if you zoom out, it should be a bit more, it should look a bit nicer. But for now, I'll just zoom in again. Maybe not this much. Okay, so from our list earlier, I remember that this tissue, this regulatory feature is active in astrocytes. So I will select that. And then you can also search for other, for cell lines. So for example, HeLa, and select that. So selecting your cell or tissue of interest will add a number to your cell tissues that you have selected. So if you click on that, oops. If you click on the plus sign here, you'll see all the different cell or tissue types that you've selected. Next, I want to show you how to turn on, how to show the different experimental evidence of this regulatory feature. So we've got experiments looking at histone, open chromatin, and transcription factors. There are several different and experiments by which you can look at the histone modifications. So you can choose to select all or just a number of them. I'll just select all for now. For open chromatin, we only have data for DNase 1 sensitivity. And then for transcription factors, we've got these two experimental ways to identify transcription factors. So on the right, again, you can see the different experiments that you've selected. And if you are happy with that, you can click Configure Track Display here on the bottom right-hand side. So now we've selected our tracks. Now we want to configure the track display. So you can see that in these steps at the top of the pop-up window. And here you can just choose how you want your data to be displayed in your view. So we've got the cell and tissue types here, and then we've got the different experimental evidence up here as columns. So these blue boxes, you can click them and you can choose to either show or to display or turn them off. You can also choose the format of which you want to show them. So you can show them as a peak and signal only, as a peak and signal, or as peak or signal only. So I'll just I want to view the entire thing. So I want to view peaks and signals. So I'll just keep that as it is at default. And if you're happy, you can click View Tracks here at the bottom right-hand side of the page. And that closes, that saves and closes the pop-up window and refreshes our view here. Okay, so it's loaded now. So we've got our tracks, we've got our regulatory build track, which we had by default already. But next, we can see information about this regulatory feature in astrocyte cells. So we can look at the different experiments that have been done to verify this. So you can click on them. You can look at them in more detail. But there's also a graphical representation of these data that we imported. So if you remember from earlier, we had those squiggly lines, and if a peak hits a certain threshold, it gets annotated on Ensembl. So this is what happened here. So for example, here we've got green represents DNase 1 enriched sites, or if you want, you can also go to the bottom of the page to check the line. Oh no, you can't. Oh, there it is. Sorry, I was looking for the legend here. So The legend you can find at the left-hand side, and then you can just see here this color is the same as this, so that's the DNase 1 sensitivity experiment that was done on this regulatory feature. Then we also got information on other experiments. These the black triangles, they just represent where these peaks have been identified. Okay, so then next we also have our HeLa cell line here. We've got the epigenome activity. If you click on this feature, it's loading right now, you can find out more information. Okay, so it took a bit of time to load, but here you can find out information. So the stable ID of this particular regulatory feature, and also all the different motif feature IDs, and the different transcription factors that bind to the site. We will close this. And then again, we have all the different experiments that were done on this cell line, and then also for the hepatocyte as well. So if you click, which I've already done, but let's just do it again. If I click on one of the experiments, you can see where or what project did this experimental verification. So in this case, it was the ENCODE project. But if you click the other features, you can find all the other different projects that have also identified regulatory features here. Or in these cases, it's mainly ENCODE, I think. Yeah. And then just wanted to say to remember that Ensembl does not link regulatory features to genes and gene expression. So we simply present you with the location of the feature, and we show you the different cell line and tissue-specific data. But it is up to you to make your own inferences. And that finishes my demonstration. So I'll head back to the presentation, and I will give you some time to try the regulation exercises. So let's do the exercises for 20 minutes, and then we'll come back and cover the last module. All right, so it's 3:40 now. I hope you had time to do the exercises on regulation. Does anyone have any questions? Okay. So I'm just reading this question here from Oge. I hope I pronounced your name right there. I'm sorry if I didn't. So he's saying there certainly will be questions after studying the documents and undertaking the exercises, and he's wondering how he can do this. So the exercises, if you look at my screen, I'm gonna open a new tab and go to training.ensembl.org. And then I will actually copy this URL, put it in the, excuse me, I'll put it in the chat box. And so this training page will be available to you indefinitely. So even after, let's say in 2 months' time, you remembered, our workshop and you wanted to try out the exercises, you will still be able to access them. This also counts for the presentation, so which you can find on this link here, and also for the living document, which is just a Google document, and that will be open for you to refer to indefinitely as well. But at the end of our workshop, so during the closing remarks, I'll also show you how you can contact us for questions and other resources where you can learn more about bioinformatics in general. But for now, I'll close this and yeah, I will go through the different resources again at the end of the workshop. Okay, so let's cover our last module of the day. I will be talking about how you can retrieve Ensembl data using BioMart. I'm going to go through what BioMart is and how to use it, and then I'll give you a brief demonstration and you'll have some time to do some exercises as well. Okay, so what is BioMart? It's a web-based tool to export Ensembl data without needing to have any programming skills. You can build queries using point and click methods, so just with your mouse, and you can generate custom data tables and files. But just remember, BioMart is not meant for downloading whole genomes. So you can go to our FTP sites for this instead. And you can use BioMart for up to 500 entries at a time. Oops, sorry. So why should you use BioMart if you can get all of the information you need from the different Ensembl pages? You can do that, but it takes a lot of time. You would have to go to the different pages, just like having to go to different specialty shops to get the groceries that you want. So to get bread, you can go to a bakery, cheese to a cheesemonger, or your greens, your vegetables at a greengrocer's. But why should you make your life difficult if you can just go to a big supermarket instead and get everything you need from a single shop? And this is what BioMart is. So it is a one-stop shop for the different data types you can get from Ensembl. On BioMart, you can query multiple genes or variants at once. You can convert IDs, extract loci, and download multiple sequences in one go. So in other words, you can use BioMart for tasks that would be too time-consuming by going through the different Ensembl browser pages alone. You can find BioMart on Ensembl and Ensembl genomes. So the structure of the browsers are the same, as you might have seen. So if you go to the top of the top of the page at the navigation bar, you can find BioMart in this location. And this goes for all the other Ensembl pages as well. So BioMart is available on the Ensembl browser, which is dedicated for vertebrate genomes, but also Ensembl plants, metazoa, protists, and fungi. For protists and fungi, BioMart is only available for a number of species. And unfortunately, BioMart is not available for bacteria, as there are too many genomes of them. All right, so now that I have introduced you to BioMart, let's look at how to use the tool. The easiest way to use BioMart is to use this 4-step principle. So step 1, choose the database and species. Step 2, narrow down the dataset. Step 3, specify your output and what to print on your table. And then step 4, choose the format of your results and export. In the first step, you are defining the database that you want to do your search in. So whether you're interested in, this is where you specify whether you're interested in genes, variation, or regulatory features, or even mouse strains. And then you choose your species of interest. In the second step, you are defining your set of genes or variants by applying different filters. So these filters can be a region in the genome, it can be a list of IDs, it can be a particular function or phenotype. So this is going to be your input. You can build simple queries by applying a single filter only. Or you can build more complex queries by applying multiple filters. So for example, let's say you have a particular region in the genome you are interested in. So that will be your first filter. But you only want to extract genes that have a certain function in that region. So the function of the gene would be your second filter. And applying these filters, these different filters, narrows down your search, leaving you with a list of genes with common features. And whatever these common features are, this is the list that we apply attributes to in the third step. In this step, you are defining the type of data you want for that set. So you are selecting the attributes of your data output. For example, you can choose if your output should have things listed like IDs, features, variants, sequences, or any homologs. And finally, once you are happy with your query, you can view and download your results. And you can get these in different formats. So you can download these in the HTML format, tab separated or comma separated format, as an Excel format. And for sequences, you can only download them in the FASTA format. Just beware when working with Excel. Sometimes Excel likes to automatically format some cells. So for example, sometimes it would change gene names to dates. Okay, so this was a brief introduction to BioMart. Let's move on to the demo. So using the 4-step system we just covered, we are going to do a query on BioMart and Ensembl. We're going to look at 6 human genes, so ESPN, MYH9, USH1C, CISD2, THRB, and BRCA2, which I'm going to copy and paste in the chat box. And we are going to find out their NCBI-IDs function and the cDNA sequence, and get the cDNA sequences. So to review, the 4 steps are select the data set. So in this case, genes and human, because we are interested in human genes. Then we apply our filters. So we are interested in a list of 6 genes. And then we choose our attributes, so what we want in our output. So we want in our output the NCBI IDs, we want the function, and we also want the sequences. And then finally, I'll show you how you can download this data and how to view it. Okay, so I'm gonna jump out of my presentation and go to my Ensembl tab. And for this, excuse me, and for this I will go back to the home page. you don't have to do that. You can use the top right-hand search box here, but I want to use the homepage, and I will paste the gene names in the middle search bar and select the species, so human, and then I will click go. Oh wait, sorry, we're doing BioMart. Ah, I was doing it automatically there. Ignore what I just said. We're not doing that. We are interested in BioMart. So to go to BioMart, I just zoomed in a bit further. To go to BioMart, you go to the top of the page and you click BioMart here. And that will send you to the BioMart tool. Okay, so this is how BioMart looks like. So we've got, if you have data already here on the left-hand side, you can just click New, which just resets all the settings, so you can start a new query from fresh. So the first step is to choose our data set. So here we click on this Choose Database drop-down menu, and we select genes because we are interested in genes. This gives us the option to select our data set, so the species. If you click the drop-down menu, you'll see that this has a list of all the species available in Ensembl. Sorry. But we are interested in human, and human is very popular, so you can find a shortcut here on top. So you click human genes, and once BioMart has loaded, you can see that we've got more options here on the left-hand side now. So now we can also apply filters. We can choose the attributes, and then we can also select another data set if we wanted to. But first, we will apply the filters. So we have a list of 6 genes. If we click filters, it will open a new section here on the right. And here you can apply different categories. So you can look at, if you're interested in a particular region of the genome, you can click the region tab here. And here you can choose, for example, particular chromosomes that you want to look at, or if you have specific coordinates, you can enter it here. You can also enter multiple coordinates in this multiple regions box here. So, for example, to add a chromosome that you want to look at, you just click on the chromosome number you're interested in, and then you'll see that now under filters, you can see that it's populated, it's added chromosome 10 to the filters. You can select multiple chromosomes by pressing the Command or Control button on your keyboard, and then just clicking on the other chromosomes you're interested in. And then you can see here again on the left, it just adds them to the list. If you want to turn a filter off, you can just click this check button, this check icon on the next to the chromosome title. Or if you just want to delete or remove one chromosome, again, you'll have to press your Command or Control button and then just click on the chromosome you want to remove. I'm going to remove all chromosomes because I'm not really interested in them at the moment. But what we are interested in are genes. So I will open this gene tab. And here we can choose different filters again. So we can limit it to certain gene lists. We can input external reference IDs, so you can click this drop-down menu, and here you can just choose one of the different IDs that you have. For example, you might have NCBI IDs, so you can select that here. You can also limit to probe sets only, and also gene types, transcript types, or sources, certain sources. So we've got Ensembl, we've got Havana, and all the other sources. And you can also select flags. So for example, if you wanted to look at main select transcripts only. But we have a list of genes, so we will need to put this into the input external references ID list section. And that's the second one from the top. So first, I'll select the type of IDs that we've got. So I'll click this drop-down menu again. And we have got a list of gene names. So what you'll do is you'll just need to search for it in this list. So gene names here, and click that. And then you paste your list of genes in this box underneath. So you can also paste them as a list. You can also paste them as a tab separated file, but I had them in a comma separated format already, so I'll just keep it this way. You can also choose to upload a list of IDs if you have one on your computer locally. Okay, so this is the gene section. We also have loads of other sections. So if you can, you can look through them in your own time. But one thing I wanted to show you is the phenotype section, because this is quite interesting. So here you have a list of different phenotypes, but unfortunately you can't search. You don't have a search function here. So what I do is, what I do instead is I use the browser's search function. So if you press Command F or Control F on your keyboard, you will have this little search option here on the top right hand side. So this is for Chrome. If you have Firefox, it will be here at the bottom. Sorry, this is the top right hand side, yeah. And if it's on Chrome, you'll have it here on the bottom left hand side. But let's say you are interested in a particular phenotype. So let's say you are a researcher in Alzheimer's disease and you want to find out information on Alzheimer's. So for example, if what genes and transcripts are located within a region, well, we select a gene that are associated with Alzheimer's disease. So you can just use the search button and then click, like in the chromosome filter option. You can click multiple ones as well if you want. That populates it here. And then if you want to remove this option, you just click on this check mark. So I just wanted to show you that in case you were interested in the phenotypes. But for our demonstration, for our purposes, we're only interested in genes. So we've got our filters. Excuse me. We've applied our filters, and now we want to move on to our attributes. So what we want to print on our in our results. Before I move on to attributes, I want to do a little sanity check. So I want to check how many genes my filters apply to. So we have a list of 6 genes, so this should only apply to 6 genes. If you press this count button here at the top of the page, this will just count the number of genes of which your filter, or if it's more complex, filters apply to. So now you can see here next to dataset, we can see that this filter applies to 6 out of 68,016 genes. So I'm happy with that. 6 genes, we have a list of 6 genes, and we'll move on to attributes. So if you click attributes, it will refresh this right-hand side with different categories now. So we have features. You can choose to show features, different structures, homologs, variants, germline variants, and somatic variants, and also sequences. So on BioMart, you can only apply one category, one attribute category at once. So we want to get the NCBI IDs and the functions, so the Go terms. So these would fall under features. If you're not sure, you can just click on the different tabs and look for them. But I know these fall under features. But we also want to get the sequences of the 6 genes. So that means we will have to do 2 queries. So we'll first get our NCBI IDs and Go terms, and then afterwards we will get our sequences by clicking this. So first, let's get our features. So I'll click on the features category, and I have the gene tab open already. And by default, you've got the gene and transcript IDs selected. And we want our NCBI IDs. So these will fall under the external tab because it's an external ID. And here we can find NCBI ID. And if you click the check button next to the name, you can see that it gets added to the attributes section here on the left. So these attributes, the order of the attributes is the same order that your columns are going to be in. So it's going to make sense once I hit results, but just remember that this order, so if you select gene transcript and then NCBI gene ID first, it'll get added as the first column is gene, second column is transcript, and the third column will be NCBI gene IDs. So if I add the Go terms here at the top of the page, if I add these attributes, these will be added at the end of the table. Okay, so we've got our NCBI gene ID, we've got Go terms. What's also good practice, we need to know the gene names. So we've applied our list of gene names, but in our results we don't actually have the names yet. So you'll need to add this separately so that we can refer our results back to our input. So the gene names is here in the second column on the right, and that gets added to the end of the list. Once you're happy with all your attributes, so everything that you want to print on your data table, you can hit results here at the top left-hand side. And that loads the results table here on the right. So by default, you will see the first 10 rows, but you can, because these will be more than 10 rows, you can click the drop-down and show all. And this just opens a new tab, giving you the full list of your results. While that's loading, I'll just show you around. I'll show you all the other options in the results page. So you can export your results too. If you click the drop down, you can choose to export it as it is, or if you like, you can also download a compressed file. And if it's quite a long, like a big query, you can choose to get your results sent to your email. So to do that, you just click this compressed web file, notified by email, and then you enter your email address under email notification. And then the next drop-down, you can choose your format. So as mentioned, HTML, comma separated, tab separated, and Excel format. And once you're happy with how you want to export your results, you just click go, and then it'll initiate the download. And so our results tab has loaded, so I'll just go back to that. And so here you can see we've got our gene stable ID, gene stable IDs, we've got our transcript IDs, then our NCBI ID, which we've selected in attributes, Go terms, and then the gene name as well. So this is what I meant with the order of, if I go back to our first tab, this is what I meant by the order of the attributes. So the order in which you added to the attributes is going to be the order of your columns. So we added gene name last, so it'll be the last column as well. Now, if you scroll down a little bit, you can see that there are multiple entries for each gene. So for example, we're here under results of BRCA2, and then I have to scroll quite far down to get to the next gene. So there are multiple entries of results per gene. And this is because a single gene can have multiple transcripts associated with it. And because a single gene can be transcribed into multiple transcripts, these multiple transcripts can also have different functions. So if you look closely, you can see that a transcript can have multiple entries as well. So let's look at, so this is the transcript ID, ends with 21618, and then the next one as well is 21618, and then the next one as well. So this is because there are multiple Go terms, which we selected here, associated with each transcript. Okay, so you can close this tab now, and we'll go back to our results. We have our list of NCBI IDs and Go terms now, and we want to get the sequences. Oh yeah, also what I just want to mention is, so I said that it's good practice to have the gene names in our attributes because we've added it to the filters. So if we didn't have our gene names here, we wouldn't know which gene this belonged to. Well, we would. We could click on each of the links, but it would be very time consuming. So to make our lives easier, I just added the gene name, a gene name column. You can also do that with regions and all the other attributes as well. Okay, so to get our sequence, we can just click back to attributes here on the left, or if you want, you can start an entire new query, but we don't need to do that. We'll click sequence on the right-hand side here and click the sequences tab. So here you can select the different sequences. We are interested in cDNA sequences. So that will be here on the right. But you might have noticed that the diagram actually changed. So depending on what you click on, it'll just show you a little representation of where these sequences are located. just as a little reminder. So we want cDNA sequences, and because this is a different category of attributes, we also need to check our headers again. So by default, we have gene stable IDs, we have transcript stable IDs. We just added cDNA sequences, but we also need the gene names so we can refer back to our input. And this is under header information. So we'll click gene name. It's here on the left column, and that gets added to the end of the list. Okay, and now you can click results again to get your sequences. And the sequences will come in FASTA format only. So this starts with a greater sign followed by the header information that we selected. So the gene stable IDs, the transcript stable IDs, and then the gene name. And again, by default, it'll show the first 10 rows. So if you wanted, you can choose to view all. I'm just going to view the first 20. And this just gives you a list of the first 20 results. So similar to before, you are going to get multiple entries for each gene. So we've got THRB here, another THRB here. And this is because you get the different transcript sequences as well. So not just the gene sequence, but also the transcript sequence. And then again, if you want to download your this FASTA file, you can click this file dropdown, select the format. So we only have FASTA here, and then click go, and that will open up your download option. Okay, so before I go back to the presentation, you can also, if you wanted to share your BioMart query with your collaborators, you can click this URL button at the top of the page. And this just gives you a static URL for you to share. Because if you just take this URL that you have in your address bar, they won't be able to see the same query that you're seeing. So it's best to share your URL by clicking this button here. Okay, so that finishes my presentation. And we now have time for some exercises. So we'll do the BioMart exercises for 15 minutes. And then afterwards, I will go through some closing remarks, also a feedback survey from where you can get an attendance certificate from as well. And then there'll be plenty of time for you to ask questions. Hi, Grace. Thank you for explaining your question. I just wanted to make sure I understood it correctly. So I will show you again how to do that. So I'll jump out of the presentation and go to the Ensembl tab. I will reset my BioMart query so you can see how it goes from the beginning. So it's just loading now. And then we chose the database, we chose the data set. And now we can select our filters. So if you click on filters on the left, you can go to region here. So that's the first tab. And then the first option that you'll see is choosing chromosomes. So if you just want a single chromosome, you just click on whatever number you're interested in. But if you wanted to select multiple ones, all you need to do is press your Command or Control button on your keyboard and then click the other chromosomes you'd like to look at. So you can see that they get added here on the left-hand side. And then again, if you want to remove them, press Command or Control button on your keyboard and then just unselect them. Okay, so I hope you had some time to do the BioMart exercises. Are there any questions before I move on with some closing remarks? Okay. All right, so um, if you do
Sorry.
have, oh, hi.
Hi, I'm Tolu. I'm a bioinformatician. I'm doing my PhD in data science. Thank you for the training. It was quite enlightening. My questions basically are: do you um have like ongoing projects and maybe possible projects that you're working on that maybe you'd like to talk about? And see how maybe there could be similar implementation of such things in our various regions. Just like want to know maybe possible projects you're working on. Yeah.
So I myself am only working in the outreach team. So we, for my case, we deliver the training only and help with questions from users. But there are a lot of other projects that Ensemble is part of. So the EBI, the European Bioinformatics Institute, has a number of these different projects. And if you like, I can share a list of some interesting ones on the living document. And you can read further on them. So for example, we didn't talk too much about them, but there is the Havana team, and they are in charge of the manual annotation of the human genome. So we have two ways of annotating the genomes. There is the automatic pipeline, but there's also the manual pipeline where actually a person goes along the genome and annotates them by hand. And I always thought that was really cool and interesting work that they were doing, because you have to be very meticulous, and it takes a lot of time to do these annotations by hand. And yeah, Alina just shared a link there with some work with collaborators as well.
Thank you. Thank you so much. Yeah, you could share the other link too. Thank you so much.
Yeah, I'll do that. Thank you. Any other questions? Okay, so thank you, everyone. You've been very great. You've been very engaging. And I just want to go through some closing notes before I let you go. So I also want to highlight that the training page and the living document will be accessible to you indefinitely. So you can go through the walkthroughs and the exercises again in your own time if you didn't have time today. And then also refer to the answers of the questions that have been asked. So again, thank you very much for participating in this workshop. And we are always eager to get feedback, which will help us improve our courses. So I think, Oge, you had a comment there and asked if we could break up these webinars in shorter chunks. So we'd be very grateful if you could give us even more feedback. Um, I understand that it is difficult to attend a day-long workshop, but there will be more opportunities, if you like, um, to attend these kind of workshops. So, oops, sorry. Um, we also do open webinar series where we actually chop up the modules in shorter timings, so on multiple days. So to complete this feedback, I will post a link to the chat box, but you can also find it in the training on the training page. So I've got a tab open already, I think. Yes. So this is the Baze University training page, and you can find the feedback survey here under survey. So if you click that, it just has a few questions and some comments that you can give us. But at the end of the page, you can choose to get an electronic certificate of attendance. And for this, you just click yes and then give your email address and name, and it will get sent to you by email. So I'll give you 5 minutes to do this, and then I'll finish up.
Okay, Dr. Louise.
Hi, Grace.
Yeah, for some people that were logged out as a result of network, is it possible to send the link for feedback to them?
Yeah, of course. So I will keep this feedback survey open for a couple days so that the other participants um, can do these as well and get the attendance certificate.
Okay. Thank you.
No problem.
And I also hope that we'll have another webinar soon. Maybe we'll get back to you.
Yeah, we're always happy to give these workshops. We want to train as many people as possible.
Okay. All right, thank you.
Thanks. Okay, thank you to those who have completed the feedback survey. We really do appreciate every and any comment so that we can improve our courses for future participants. Okay, so before I let you go, I just want to finish up with some closing words. So I want to summarize what we've learned about Ensembl today. We know that Ensembl is a genome browser that integrates different types of data. Alina gave you an overview of the website and how to view gene annotations. We learned how to retrieve variation data, how to view the comparative genomics data available in the browser, and where to find regulation data. And finally, in our last module, I showed you how to retrieve large sets of Ensembl data using BioMart. We know that we can access data in Ensembl in different ways. We can go to the Ensembl browser sites, which was the main focus of this workshop, but you can also retrieve large amounts of data through the BioMart tool, which again, we've covered. But also you can access whole genome sequence data through the FTP sites or programmatically using API or MySQL. If you enjoyed the training, then please recommend us to your colleagues. Apart from the Ensembl browser workshop, we also have the REST API workshop, and we also offer a Train the Trainer course. And if you're interested in organizing a workshop, as Grace did for this, please contact us on the website, or you can contact us. You can find out more information on the website here, or you can email us directly under helpdesk@ensembl.org. And there are several ways to get help for Ensembl. So I mentioned the email address, but you can also go through online courses in your own time. So especially for those who didn't get a chance to stay for the entire day, there are many general bioinformatics introductory courses. So you can watch Ensembl walkthrough videos by visiting YouTube. You can view tutorials on the EBI course website, or on the Ensembl website for Ensembl-related tutorials. And you can email us, and you can also sign up to our mailing lists, through which we update our users on any news of Ensembl. And then you can also follow us on our social media. So we are on Facebook, Twitter, and LinkedIn, and we regularly share updates, or you can read our blog.
So I can snap and send to those people.
Hi, do you have a question? Okay, I think that was just an accident. But you can follow us on Twitter, Facebook, and LinkedIn. We have a blog as well where you can read about any updates of Ensembl. We post job ads there as well, and we also have a blog series where we interview a member from Ensembl and get to know them a bit better. And so Ensembl is completely open source, so we appreciate if you could cite us whenever you use Ensembl in your work. And it will help us to track who's using our resources and what kind of research you do. And with that, I just want to point out that the entire Ensembl team is not just Alina and myself and our manager Ben, but we are a team of about 100 people, and it is a massive team effort to make this project possible. And with that, I'd like to thank you for participating in the Ensembl workshop. I want to thank Baze University and Grace for hosting us, and every single person in the Ensembl team and our funding bodies. And I just saw a question from Murtala about the training page URL. So I will just copy that again and put that in the chat box for you. All right, so thank you very much. If you have any more questions, Alina and I will stay for a couple more minutes for you to ask questions.